Good morning and welcome. Ms. Bata.
Good morning. This is Lynn Bata. I am the immunization clinical consultant for the Department of Health, and I have no conflicts.
Good morning. Welcome. Dr. Bell.
Beth Bell, clinical professor, Department of Global Health, University of Washington, and I have no conflicts.
Welcome. Good morning. Dr. Bernstein.
Hello, everyone. I'm Hank Bernstein. I'm a professor of pediatrics at the Zucker School of Medicine at Hofstra Northwell, and I have no conflict.
Good morning. Welcome. Dr. Chen.
Wilbur Chen, professor of medicine at the University of Maryland School of Medicine's Center for Vaccine Development and Global Health. I have no conflicts.
Welcome. Good morning. Dr. Daly.
Morning. Matt Daly. I'm a senior investigator at the Institute for Health Research at Kaiser Permanente, Colorado, and also an associate professor of pediatrics at the University of Colorado School of Medicine. I have no conflicts of interest.
Welcome and good morning. Dr. Fry.
Good morning. Sharon Fry. I am a professor of internal medicine at St. Louis University and the clinical director for the St. Louis University Center for Vaccine Development. I do have conflicts, and they include being the St. Louis University site principal investigator for the phase 3 Moderna COVID vaccine trial and the Janssen COVID vaccine trial. Thank you.
Thank you. Good morning. Welcome. Dr. Cotton.
Neil Cotton. I am the clinical director of compromised host infections at Massachusetts General Hospital, and I am an associate professor of medicine at Harvard Medical School. I have no conflict.
Thank you. Good morning and welcome. Our Vice Chair, Dr. Lee.
Thank you. Good morning. This is Grace Lee. I am professor of pediatrics at Stanford University School of Medicine, CMO at Stanford Children's Health, and I have no conflicts.
Thank you. Good morning. Welcome. Dr. Long.
Hello, I'm Sarah Long. I'm professor of pediatrics at Drexel University College of Medicine in pediatric infectious diseases in Philadelphia, and I have no conflict.
Welcome. Good morning. Ms. McNally.
Good morning. Veronica McNally, president of the Bernie Strong Foundation based in Michigan, and I have no conflict.
Morning. Welcome. Thank you. Dr. Paling.
Good morning. Kathy Paling, professor of pediatrics and epidemiology and prevention at Wake Forest School of Medicine. I have no conflicts.
Welcome. Good morning. Dr. Sanchez.
Good morning. This is Pablo Sanchez. I'm a professor of pediatrics and neonatology and pediatric infectious disease specialist at Nationwide Children's, the Ohio State University in Columbus, Ohio. Thank you.
Thank you. Good morning.
I have no conflict.
Thank you. Dr. Talbot.
Good morning. I'm Kip Talbot. I'm an associate professor of medicine. I do adult infectious diseases at Vanderbilt University Medical Center, and I have no conflicts of interest.
Thank you. Welcome. So everyone is present here from the ACIP voting members. I will now go on to the ex officio representatives. I will call your organizations. Please state who you are and you're present. Centers for Disease Control and Prevention.
Good morning. I'm Melinda Wharton from the Immunization Services Division at CDC.
Thank you very much. Centers for Medicare, Medicaid Services.
Good morning. This is Mary Beth Hans from CMS.
Thank you. Welcome. Food and Drug Administration.
Good morning. Doran Fink, Food and Drug Administration, Office of Vaccines.
Welcome. Health Resources and Services Administration.
Captain Dale Mishler, standing in for Dr. Mary Rubin.
Morning. Welcome. Indian Health Service.
Good morning, Captain Tom Weiser for Indian Health Service.
Good morning. National Institutes of Health.
Good morning, John Weigel, National Institute of Allergy and Infectious Disease, NIH.
Welcome. Office of Infectious Diseases and HIV/AIDS Policy.
David Kim, representing OIDP.
Thank you all. I will now turn to the liaison representatives, calling each liaison group. Please state your name and any conflict of interest. American Academy of Family Physicians.
Pamela Rockwell, AAFP, no conflict.
Thank you. Welcome. American Academy of Pediatrics.
Good morning. Bonnie Maldonado, Professor of Global Health and Infectious Diseases, Stanford University, and Chair of the Committee on Infectious Diseases for the American Academy of Pediatrics.
Thank you. American Academy of Pediatrics Red Book.
David Kimberlin, University of Alabama at Birmingham, Pediatric Infectious Diseases. I'm editor of the Red Book.
Thank you. Welcome. American Academy of Physicians Assistants.
Marie-Michelle Leger, Director of Clinical Education, American Academy of PAs, no conflict.
Good morning. Welcome. American College of Health Association. Sorry, American College of Health Association.
Good morning, Sharon McMullen, Cornell University, and signing in for my colleague, Dr. K.V. Chai, University of North Carolina at Chapel Hill. Neither of us have conflicts.
Welcome. American College of Nurse Midwives. American College of Nurse Midwives. Okay. Passing out to the next group. American College of Obstetricians and Gynecologists.
Good morning. This is Dr. Linda Eckert. I'm a professor of obstetrics and gynecology at the University of Washington in Seattle, and I am the liaison for ACOG. Thank you.
Thank you. Welcome. American College of Physicians.
Good morning, Dr. Jason Goldman, General Internal Medicine, Affiliate Professor, Florida Atlantic University, representing the American College of Physicians. Always a pleasure to be here. Thank you.
Welcome and good morning. American Geriatric Society.
Morning, Ken Spader for AGS.
Good morning. America's Health Insurance Plans.
Yes, this is Bob Gluckman. I'm Chief Medical Officer for Providence Health Plans. I do have some minor stock holdings that are unrelated to any votes that may occur today. I'm a non-voting member, but I have minor stock holdings in Abbott, AbbVie, Walgreens, and Bristol Myers.
Okay. Thank you. American Immunization Registry Association.
Good morning, Rebecca Coyle representing ERA.
Thank you. American Medical Association.
Sandra Freyhofer, practicing general internist in Atlanta, and adjunct associate professor of medicine at Emory, representing the American Medical Association.
Thank you. Welcome. American Nurses Association. American Nurses Association. Okay, we'll come back. American Osteopathic Association.
Yeah, Stan Grau, Professor Emeritus at Oklahoma State University, representing the American Osteopathic Association. Present, thank you.
Thank you. Welcome. American Pharmacists Association.
Good morning. This is Steve Foster. I'm here.
Good morning. Association of Immunization Managers. Association of Immunization Managers. All right, we'll come back. Association for Prevention, Teaching, and Research.
This is Paul McKinney, Professor, School of Public Health and Information Sciences, University of Louisville.
Welcome, thank you. Association of State and Territorial Health Officers.
Good morning, Dr. Romero. This is Nirav Shah. I'm the director of the State of Maine Center for Disease Control and also the president of ASTO. I have no conflicts. Thank you.
Good morning, welcome. Biotechnology Innovation Organization.
Phyllis Arthur on behalf of BIO. Good afternoon.
Good morning, thank you. Council of State and Territorial Epidemiologists. Council of State and Territorial Epidemiologists. Okay, we'll come back to you. Canadian National Advisory Committee on Immunization. Canadian National Advisory Committee on Immunization.
Good morning. Can you hear me?
Yes, we can. Thank you.
Hi, good morning. It's Althea House representing the National Advisory Committee on Immunization for Canada.
Good morning. Infectious Diseases Society of America.
Good morning. This is Jeff Duchin, Professor in Medicine, Division of Infectious Diseases at the University of Washington in Seattle, and Health Officer for Public Health, Seattle and King County, representing the Infectious Diseases Society of America. I have no conflicts.
Thank you. Good morning. International Society for Travel Medicine.
Good morning. Elizabeth Barnett on behalf of the ISTL.
Good morning. Thank you. National Association of County and City Health Officers, or officials, excuse me.
Hey, good morning. This is Matt Zahn representing NACHO. I have no conflicts.
Thank you. Good morning. National Association of Pediatric Nurse Practitioners.
Good morning. Patsy Stinchfield from Children's Hospitals and Clinics, Minnesota, nurse practitioner in infectious disease and immunology, representing NAPNAP, no conflicts.
Thank you. Welcome. National Foundation for Infectious Diseases.
This is Bill Schaffner, Professor of Preventive Medicine and Infectious Diseases at Vanderbilt and Medical Director of the NFID. No conflicts. Good morning.
Good morning, sir. Welcome. National Medical Association.
Good morning. This is Patricia Whitley-Williams, Professor and Chief of Pediatric Infectious Diseases at Rutgers Robert Johnson Medical School, representing the National Medical Association. Thank you.
Thank you. Good morning. Welcome. Pediatric Infectious Diseases Society.
Yeah, this is Sean O'Leary, Professor of Pediatrics at the University of Colorado and Children's Hospital Colorado, representing the Pediatric Infectious Diseases Society, and I have no conflicts.
Good morning. Welcome. Pharmaceutical Research and Manufacturers of America.
Good morning. Corey Robertson, President.
Thank you. Welcome. Society for Adolescent Health and Medicine.
Hi, it's Amy Middleman. Good morning. I'm representing SAM today.
Welcome. Society for Healthcare Epidemiology of America.
Good morning. This is Marcy Dries, Chief Infection Prevention Officer and Hospital Epidemiologist at Christian Care and Associate Professor of Medicine at Sidney Kimmel Medical College at Thomas Jefferson University, and I have no conflict. Representing Shay. Thank you.
Thank you. Good morning. I will go back to those who did not respond. American College of Nurse Midwives. All right, one more time. American College of Nurse Midwives. Okay. American Nurses Association.
Good morning. This is Chad Riddle, representing the ANA.
Good morning. Welcome. The Association of Immunization Managers. Association of Immunization Managers. Okay. Moving on. Council of State and Territorial Epidemiologists. So apparently, Susan Lett, Dr. Lett is on the line, but we're not able to hear her, so we'll mark her as present. Very good. So that completes our roll call. Let me see here. We'll move on then, I think, to our first topic, which is coronavirus disease 2019 vaccines, with an introduction by the COVID-19 Workgroup Chair, Dr. Bell. Dr. Bell, are you ready?
Yes, Dr. Romero, here I am.
Thank you. Go ahead, please.
Thank you, and good morning to everyone. Could I have the next slide, please? It wasn't very long ago that we last met, and so I'm just going to spend a minute or two going through some of the details of what we've been up to before the last emergency meeting and since then. So during the week of April 12th, as many of you are already aware, on Monday there was a meeting of the Vaccine Safety Technical Group, followed on Tuesday by a joint CDC-FDA statement, which recommended a pause in the use of the J&J vaccine, and a Health Alert Network notification was released. Also on Tuesday, the ACIP COVID-19 vaccines work group met, And on Wednesday, April 14th, there was an emergency ACIP meeting. Now, this emergency meeting was meant to consider the implications of reported cases of thrombosis and thrombocytopenia after the Janssen J&J vaccine on vaccination policy. And after discussions regarding the U.S. cases of central venous sinus thrombosis and thrombocytopenia, the ACIP agreed that more information was needed before policy recommendations could be made regarding the Janssen vaccine. Next, please. So during the week of April 19th, on Monday, the VAST met again. On Wednesday, there was an additional special meeting of the ACIP COVID-19 vaccines work group. And on Thursday was a second meeting of the ACIP COVID-19 vaccines work group. Today, we are meeting again in emergency ACIP session. And the purpose of this meeting is to review the updated cases of thrombosis with thrombocytopenia after the Janssen COVID-19 vaccine, to discuss the risk-benefit analysis and policy options for updated recommendations for use of this vaccine. Next. Today's agenda, I'll just go over that very briefly. First, we're going to hear from Dr. Michael Streif from the Johns Hopkins University, who will present about the pathogenesis and management of thrombosis with thrombocytopenia syndrome. And then Dr. Tom Srinivakuru from CDC will talk about the thrombosis with thrombocytopenia syndrome, TTS, following the Janssen COVID-19 vaccine. We will have a public comment period. Then we will hear from the manufacturers of the Janssen vaccine, Dr. Matai Mamin, who is the director of the global head for Janssen Research and Development, and Dr. Joanne Waldstreicher, who is the chief medical officer of J&J. Dr. Grace Lee, the co-chair of VAST and ACIP member, will give the VAST assessment. Then we will hear from Dr. Oliver with a presentation about applying the evidence to recommendation framework to the TTS after COVID-19, the typographical error there, vaccines. We will have a discussion period and then a vote on updated interim recommendations for use of the Janssen COVID-19 vaccine. Next. Again, I just want to acknowledge all of the work group members, ex officios, liaisons, consultants, and the CDC lead, Dr. Sarah Oliver. Next. As well as the large number of CDC participants who have been working very hard to put together the information that we will be hearing today. Thank you. I think that's it for me. And I'll turn the gavel back to you, Dr. Romero.
Thank you, Dr. Bell. So we'll move on to our next presentation, which is the pathogenesis and management of thrombosis with thrombocytopenia syndrome, TTS. We will ask Dr. Michael Streif to please go forward. And forgive me if I mispronounce your name.
Oh, no, you did fine. And hey, this is Mike Streif, and I appreciate the invitation to present today. Next slide. So this is just for your review, disclosures. I have done some consulting for these companies, specifically on development of anticoagulant medications and for coagulation testing, as well as some research support from the funders you see on the slide. Next slide. So I thought first, before we get into the TTS syndrome, that I'd discuss a little bit about cerebral venous sinus thrombosis because it's an unusual thrombotic event that compared to what we usually see in clinical practice, which much more common events would be deep vein thrombosis and pulmonary embolism. But it's similar in the fact that it's a thrombotic event that occurs in the deep veins or superficial veins of the brain, as you can see in this nice picture there. The incidence, it's quite a rare event that there are about 10 to 15 cases per million in the population. So this is far rarer than when with deep vein thrombosis, pulmonary embolism, we see 1 in 1,000 in the general population on an annual basis. The mean age for patients with this is about 35 years of age, so a younger patient population than typical for other venous events. And it tends to occur more frequently in women than men by about a 2-to-1 ratio. Typical presentation for patients is headache. Vast majority of patients have a headache. More severely affected patients can have seizures related to the injury, limb weakness, or reduced level of consciousness, or even coma. Risk factors for CVST in the general population are use of estrogen-containing oral contraceptives, clotting disorders, pregnancy, cancers, particularly of the central nervous system, or infections of that location, or surgeries, particularly of that location. Generally, the diagnosis is made with either a contrast CT scan of the brain or of the or magnetic resonance imaging focusing on the veins. And treatment generally is anticoagulation. In extreme cases, thrombolysis or clot-busting medications or devices will be used. Next slide. So what are the clinical characteristics of the thrombosis with thrombocytopenia syndrome so far? And this is, I'd say, a moving target because we're still early in the acquisition of cases. But so far, it appears to be a thrombotic response to receiving an adenoviral vector vaccine against SARS-CoV-2. The incidence, and this is based on the European experience and the United Kingdom experience, is anywhere from 1 case per 100,000 to 1 in 250,000 of vaccine recipients. This is a moving target, still being, I wouldn't consider this gospel or accurate data. Age range, very broad, anywhere from 20s to as old as 77 years of age, although the vast majority of patients have been less than 60 years of age. Generally affects women more than men. Immediate onset of the symptoms after vaccination is about 9 to 10 days. This is looking at the European and UK data with the range up to 24 days after vaccination. The most common thrombotic events are cerebral venous sinus thrombosis, although some patients have also had DVT or PE. A couple of patients have presented with just deep vein thrombosis or pulmonary embolism and not CVST. There have also been abdominal vein blood clots, and some patients have had arterial thrombotic events. What's unique about this compared to other thrombotic events is that these patients tend to have positive tests for an entity known as heparin-induced thrombocytopenia, which is a reasonably infrequent adverse event to receiving particularly unfractionated heparin therapy. And when this testing has been done on the patients that have been reported so far, they seem to be uniformly positive for the platelet factor 4 heparin antibody immunoassay. And this is in particular an ELISA assay. Some of the other immunoassays have been negative, rarely, although we have a small experience, I emphasize. And platelet activation assays that are typically used for diagnosing this entity have been positive in some cases, although you'll see in the U.S. cases not uniformly positive. Platelet nadirs generally in the 20,000 range. As you can see, the range there anywhere from 7 to 113,000. Fibrinogen values tend to be low. Normal range for fibrinogen is usually 200 to 250 to 400 to 450 milligrams per deciliter. Some patients, a rare patient had an elevated fibrinogen, but most patients tend to be normal or low. D-dimer levels tend to be markedly elevated. reflecting a thrombotic process. And these patients clinically, when they've been treated with unfractionated heparin upon presentation, they've had progression of their thrombotic events. And then platelet recovery tends to occur with use of a non-heparin anticoagulant as well as intravenous immunoglobulin. Next slide. So this is just a brief comparison of the two vaccines and the characteristics. And as you can see, comparing one side to the other, the age range is similar between the two vaccines, although there have been many more cases with the Oxford AstraZeneca vaccine. The number of cases, as far as gender distribution, more frequent with women than men. And the onset about the same, that anywhere from 5 days to up to 3 weeks or so. Presentations have been similar since most patients are presenting with a cerebral venous sinus thrombosis. Headache is prominent, although abdominal pain, back pain is also prominent, and then signs of stroke, so leg or arm weakness or disturbances in consciousness. Thrombotic events, as you can see, have been in similar locations, as have platelet nadirs. The heparin platelet factor 4 assay, the immunoassay, that is the ELISA assay, has been uniformly positive. Platelet function or functional platelet activation assays have been, in the European experience, positive, less positive in the U.S. experience using a different assay, the serotonin release assay. Next slide. So this is a cartoon that attempts to show how we think this, or what's going on in this syndrome. How are these thrombotic events developing? Why do these patients have thrombocytopenia? And this is based on the published work so far, primarily from Europe, Greinacher, as well as Schultz, but primarily Andreas Greinacher, who spent his whole career studying heparin-induced thrombocytopenia. And so a lot of this understanding of what heparin-induced thrombocytopenia is based on his work. What you can see in this cartoon is you see first on the left side of the cartoon, you have a platelet with an alpha granule. Inside alpha granules is a protein called platelet factor 4, also shown as PF4. And that is a chemokine that's important for controlling, it's, I guess, a suppressor of endogenous anticoagulants like heparan sulfate, also is involved in the wound healing process. So in patients that have a lot of activation of platelets, you'll release a lot of platelet factor 4 to help in the thrombotic and healing process. If someone has been exposed to a polyanion, such as unfractionated heparin, these complexes of platelet factor 4 and heparin can form, as you see in the top end of this diagram. And then what this does is converts platelet factor 4, a portion of it, into an immunogen that the immune system responds to in a small percentage of patients. And so what happens is they develop antibodies against this neoantigen on platelet factor 4 that's generated by the interaction between the polyanion heparin and platelet factor 4. And then these antibodies can bind to the Fc receptors on platelets. And then what this results in is profound platelet activation. And so you get consumptive thrombocytopenia in this situation where platelets are being activated and form lots of platelet microparticles, as you can see toward the bottom right-hand side of the slide, where there's lots of these little platelet microparticles that increase the surface area of platelets. And also because they're activated, they have a lot of phosphatidylserine exposed, which serves as kind of a playground for the clotting factors and activates the coagulation cascade. And so what you generate is a vicious cycle, a thrombotic storm that consumes more platelets, activates more platelets, and causes clotting. Next slide. Platelets, now this is a beautiful diagram that's horribly complex, but I couldn't find a better one in the literature that kind of outlines that it's not just platelets that are activated by antibodies that are involved in heparin-induced thrombocytopenia. The endothelium is activated. It expresses tissue factor. It's also injured, releasing von Willebrand factor that can cause platelets to aggregate. Monocytes, one of the white cells, are activated, and they express tissue factor as well. Again, that activates the coagulation cascade. And then finally, neutrophils are activated and express these in their activation nets, or neutrophil extracellular traps, that, again, form a place where you can activate platelets, you can activate the coagulation cascade. Now, what I've discussed so far focuses completely on heparin because that's the most common trigger for this syndrome. But there's a variant of it that a small number of patients have been reported that develop something called autoimmune thrombocytopenic purpura, which is where the patients have not received heparin at all, and yet they develop the syndrome. And this has been seen in people that have had surgery, and it's thought that endogenous polyanions like heparan sulfate or chondroitin sulfate or even DNA RNA complexes can bind to platelet factor 4 and generate these antibodies. And this is what is thought to be the pathogenesis that we're seeing with the vaccine, based on primarily on Andreas Greinacher's studies that are published in the New England Journal. Next slide. And so this is kind of the data that, kind of a compilation of the data that supports the current, concept of what is causing this syndrome, that it appears to be, based on laboratory testing and clinical presentation, similar to a case of autoimmune heparin-induced thrombocytopenia, because none of these patients have been exposed to heparin, yet they develop a syndrome that looks very much like it. What supports that? Well, the laboratory testing is consistent with autoimmune HIT, that they have positive platelet factor 4 immunoassays, and certainly in Greinacher's lab with his platelet activation assay, they are also uniformly positive. If you add platelet factor 4 to the reaction mixture, it amplifies the reaction, which is typical of autoimmune HIT. You can also inhibit platelet activation in vitro with either high concentrations of heparin that bind or that block the formation of the multimolecular complexes that activate platelets, or you can block it also with intravenous immunoglobulin, which is why it's used for the treatment of this syndrome. The clinical course also mirrors it, that it's very typical of autoimmune thrombocytopenia, where patients come in with thrombocytopenia and a thrombotic episode that is resistant and actually is promoted by heparin treatments. And it improves when you use a non-heparin anticoagulant as well as an intravenous immunoglobulin. Now, the etiology of this is still very unknown, I would say. It's unclear. Dr. Greinacher has published something that's on Research Square, not yet peer-reviewed, where he looked at patients that had SARS-CoV-2 infection and did analysis of platelet factor 4 structure as well as the spike protein, could not find any patients with SARS-CoV-2 infection that had platelet activating antibodies. So he doesn't think, based on his research, that there's molecular mimicry like we see with HIV and ITP. We don't have any data for that. So I think still it's unclear what causes this to develop. Next slide. So the management, and this is based largely on the ASH guidelines, although ISTH and Anticoagulation Forum also have recommendations in this regard, that for clinicians, we need to maintain a high index of suspicion that if anyone presents with any thrombotic event associated with thrombocytopenia, we have to consider this syndrome. Then you have to confirm the thrombotic event with imaging, and then at the same time, you're simultaneously testing for heparin-induced thrombocytopenia with a platelet factor 4 immunoassay, and then following those positive test results up with an activation of functional assay. Of course, the physicians should consult a hematologist so they can confirm the diagnosis, also rule out other diagnoses that can present similar to this thrombocytopenia thrombotic syndrome, and then treat with a non-heparin anticoagulant, and then treat the thrombocytopenia with intravenous immunoglobulin, which blocks antibody activation of platelets and speeds recovery of platelets, and avoid platelet transfusions because that seems to feed the fire of this syndrome. Next slide. So with that, it kind of concludes my remarks. I'd be happy to take any questions. Thank you very much for the invitation again.
Thank you very much for that outstanding review. We are now open for questions. I'm looking for hands, just a minute. Dr. Lee, please.
Thank you so much for that excellent presentation. I wanted to ask about your comment about, well, in some reports, some have noted that the depth of thrombocytopenia might be associated with worse outcome. So I guess my question is, does that reflect how long the process has been going on? And is it possible that if IVIG were administered early, that could impact the overall outcome of patients?
So very good question. I would say that the depth of thrombocytopenia could suggest that the process has been ongoing longer, or should have some, maybe reflects the titer of antibodies, the immune reaction that's been triggered. It could be either one of those things. And I think that in those patients, you really do need to use intravenous immunoglobulin. We don't always use IVIG for typical heparin-induced thrombocytopenia patient treatment, but we do use it in people that have severe syndromes. And these patients seem to have a very severe clinical presentation. And so I think IVIG should be used if it's available for their treatment.
Thank you. Dr. Sanchez.
Thank you. That was a very nice explanation. I was just wondering, because the role of EDTA, at least in the AstraZeneca vaccine, has also been implicated to cause some vascular permeability, and I was wondering where that may fit into all of this, looking at other constituents of the vaccine.
I have no idea. I can't see how that might trigger the antigen unless it's causing release of, there is heparan sulfate, dermatan sulfate bound to the endothelium. If the vaccine is triggering release of those compounds in higher proportions so that they can bind to platelet factor 4, and the vaccine is also activating platelets, I can see you, but I'm doing a lot of hand waving. You can't see it, but that's what's happening. But I don't know. I mean, I think that still remains to be seen and tested as a hypothesis.
Thank you.
Sure. Sorry I didn't have a better answer.
Dr. Bernstein.
Fascinating presentation. Thank you. I was wondering, you listed 6 risk factors associated with TTS. Would screening for those risk factors eliminate those people getting this vaccine and therefore decrease the incidence of TTS after vaccine?
Unfortunately, I don't think so. That if you basically, I reported like comorbid conditions or treatments that some patients that have been reported have had, but we've also had patients that were not taking oral contraceptives or hormonal therapy, were not obese, and there's no, a patient or two have had thrombophilic or clotting states, but the vast majority have not had any of those associations. So I think it's, I guess the major way we could focus on patients on who are at risk, or it seems like it affects people that are younger in age and seems to affect women more than men so far. But I don't think we can just focus on oral contraceptive users or obese patients and as far as for vaccine, to exclude them from vaccine, getting the J&J vaccine or the AstraZeneca vaccine. I don't think we can do that.
Thank you.
Sure.
Dr. Daly.
Thanks for that excellent presentation. To what extent do you think treatment is improving outcomes, or sort of what's on the horizon in terms of treatment? Thank you.
So I think that recognition that this syndrome exists is helping to improve outcomes. I think that recognition among physicians, also recognition in the public that if you develop a severe headache, severe abdominal pain, that you really need to see your doctor and see someone for further evaluation. And I think those two pieces, will improve outcomes, because it's clear that at least in some of the cases where people, the published cases, they tried to treat their symptoms at home for a number of days before they presented, and when they presented, they were severely ill. So I think that early presentation by education of public and physicians will improve outcomes. As far as other medications, I think right now we're stuck with what we have, although I think educating doctors to avoid heparin therapy in these patients will improve outcomes. That's another thing that will improve outcomes, and using a non-heparin anticoagulant and thinking about intravenous immunoglobulin early, particularly in someone that's severely affected, that will help improve outcomes. So I think with education, we can improve outcomes of these patients.
Thank you. Before I turn to the liaison members, are there any more questions from the voting ACIP members?
Dr. Romero, this is Sarah Long. I don't seem to be able to get my hand raised to work.
Please go ahead,
May I
Dr.
ask
Long.
a question?
Of course, please.
You quoted a number for the occurrence of cerebral venous sinus thrombosis of 1 in 10 to 15 million cases. 10 to 15.
Oh, so yeah, so 1 in 100,000, yeah.
That was with vaccine. I'm really talking about if we're talking about what the population range of cerebral venous thrombosis is, was that without thrombocytopenia? And do you have any clue that what the number might be without vaccine for TTS?
Very good question. So in the first slide, I quoted, the published studies suggest that the incidence of cerebral venous sinus thrombosis is about 1 in 100,000, or it's 10 per million, roughly. But as far as people presenting with thrombocytopenia, that is extraordinarily rare in my experience. And I have not seen that reported in published literature for cerebral venous sinus thrombosis. That generally is not a feature of it. So the thrombocytopenia in association with these clotting events is unique. I couldn't give you a number on how many people with cerebral venous sinus thrombosis have thrombocytopenia on presentation. I can just tell you from my experience treating these patients, I've just never seen it. It's just not, you typically have a clot and the platelets are in the normal range. The incidence with the vaccines is based on estimates I saw in one of the editorials that had looked at the number of vaccines delivered, and this is European data, number of vaccines delivered in Europe and UK, and they estimated between 1 in 100,000 and 1 in 250,000 vaccinated patients.
Thank you.
Yeah, sure.
Thank you. Just a note, Dr. Oliver has some numbers that she'll share later that might clarify that question a little bit more. Dr. Long, did you have additional questions?
No.
Very good. Anyone else from the... Yes, who is this?
This is Kathy Failing.
Yes, are you having problems? Oh, there you are. I see you. Yes, please, go ahead.
So, Dr. Stripe, thank you very much for a very informative presentation. If I understand correctly, there's a female predominance for cerebral venous sinus thrombosis in its traditional presentation. There's a female predominance for the thrombocytosis and thrombocytopenia associated with adenoviruses. Now, when we get to the autoimmune form of this syndrome with heparin or other, is there a female predominance for that as well?
So for heparin-induced thrombocytopenia, there generally, not a particular predominance between men and women. Maybe slightly more women than men, but not there's not a dramatic difference as far as for developing heparin-induced thrombocytopenia. As far as for autoimmune hit, it's a rare bird, so I don't have a good idea as to what the gender breakdown is for autoimmune hit, which is a much less common form of heparin-induced thrombocytopenia.
Thank you. Dr. Bernstein, is your hand up again, or is that a leftover?
Leftovers, my apologies.
No, that's fine. I'm just making sure. Ms. Stinchfield.
Thank you, Dr. Straits. Very helpful presentation. To your point about clinicians thinking about using IVIG early, do you have any information about our IVIG supply in the U.S. right now? You know, over the years, we've had times where we were quite low in ability to get and use IVIG. Any information on that?
Unfortunately, I do not. I don't have that. And I guess if clinicians do not have availability of IVIG in their location, I would start a non-heparin anticoagulant, which is typical of what we've done for years with heparin-induced thrombocytopenia, and see if they couldn't order some as early as possible or get them to a medical center where they might be able to get it. But I don't have an idea what our supply is for IVIG. No.
Thank you.
Sure.
Thank you. Dr. Dushin.
Thank you. Thank you for your presentation. I'm sorry if I missed this, but is there a difference in either severity of the syndrome or clinical outcomes, can you say, between cerebral venous sinus thrombosis with and without thrombosis with thrombocytopenia syndrome?
It's as far as, so I think, just to clarify your question, you mean is there a difference in outcomes between people that develop this TTS syndrome versus the typical cerebral venous sinus thrombosis?
Yes.
I think that it seems like these patients that develop TTS have a more severe, their outcomes are worse. But that may be due to the fact that initially some of these patients have been treated with heparin, which may promote further, progression. So that's an added wrinkle that may make it so that they don't get the right anticoagulant treatment, they don't get the IVIG in as timely a fashion, because you don't have to worry about that with CVST. You just treat them with heparin. So they'll get treatment right away. So I think so far the outcomes have tended to be worse, but that may be because the treatment is more you have to have this specific treatment. And again, education may help with that. I think getting you to present earlier, and then doctors implementing the right management strategy may reduce, may make their outcomes more closely parallel typical CVST patients.
Thank you.
Sure.
Dr. Sanchez?
Yeah, I had another question. So you're saying that those who present with cerebral venous sinus thrombosis in general don't present initially with thrombocytopenia?
Yes.
Okay. Because there's animal study in mice where adenovirus associated thrombosis has been initially seen with, they do cause thrombocytopenia. So I'm trying to delineate what it is in these vaccines. Something about the vector may be, I'm just speculating, but there have been, some adenovirus-induced thrombocytopenia that's been seen in mice, and so, which makes it then different than what has been seen with the non-vaccine associated cerebral venous thrombosis.
I think you're right. That is a very it's a hypothesis that may be why there's differences between typical cerebral venous sinus thrombosis and this TTS syndrome. That but I don't know of any data that we have that the vaccine is activating platelets, and then somehow there's a endogenous polyanion that's forming these platelet factor 4 antibodies, but it could be, and that could be why it's different. I think that's probably being explored by many investigators. So, but there's got to be something different about it compared to our typical CVST patient.
Well, thank you. I agree.
No, it's fascinating. I think a great idea.
We really need that information.
Yep. No, I agree. That's a great idea.
So let me offer some comment here, and be careful when a pediatric infectious disease specialist offers comment on a hematologic condition. But my understanding from reading the literature from the German group is that not all of the vaccine enters the cell, and some of it spills over into the bloodstream. And in breaking down in the bloodstream, it's the DNA contained within the adenovirus that sets up the anion and then attracts the platelets. So, I mean, that's the theory that I have, and I don't know if that answers your question, Pablo, but I would just throw that out. Dr. Long.
Yeah, no, I think that. Oh, sorry. Yeah, I did see that Greinacher has said that's another possible avenue.
Thank you.
To these antibodies. Yes, you're correct.
Thank you. Dr. Long?
Yes, I found my hand raiser, so now you're all in trouble. I wondered if you might also give an opinion on the relative outcomes of patients who have the true heparin-induced syndrome appropriately treated compared with the vaccine-induced TTS, or what we presume is vaccine-induced TTS.
Good question. Generally, patients with the typical heparin-induced thrombocytopenia, I think have better outcomes than we're seeing with the TTS. And I think that the primary reason for that is the location of the clot. That I can't think of a patient that I've managed with heparin-induced thrombocytopenia where they've developed cerebral venous sinus thrombosis. And typically they develop deep vein thrombosis or pulmonary embolism or an arterial clot. But having clots in the CNS and then secondary intracranial hemorrhage, it's just a bad place to have a clot. And so that's why I think the fatality rate of this syndrome or the rate of people having permanent neurologic or at least ongoing neurologic problems is much higher than we see with typical HIT patients because they're not clotting in their brain.
Thank you.
Dr. Gluckman.
Sorry, it took me a while to get unmuted. Can you give us some sense of which the incidence of thrombotic events with complications that are actually secondary to COVID and how that compares to the complications related to the vaccine?
Yeah, so I'll give you what this may be in other talks too. There is an article out. I don't know if it's in Research Square. I saw a preprint of an article from Oxford, I believe, that looked at specifically cerebral venous sinus thrombosis in patients that had COVID, and this is all based on administrative data, so observational data. We don't have data that they had this syndrome, the patients with COVID, but that cerebral venous sinus thrombosis appeared to be about 10-fold more common than we're seeing it in the patients that are getting the vaccine-associated cerebral venous sinus thrombosis. And I had not heard that before. This is, I think, a brand new analysis that was done with administrative data because of this vaccine-associated syndrome that is developed. So, and of course, we know from all the published reports on COVID, there is deep vein thrombosis and pulmonary embolism is not an uncommon event in critically ill patients with COVID, that 5% to 10% of critically ill patients will develop a DVT or pulmonary embolism during their COVID infection.
Dr. Shaw.
Thank you.
Thank you. This is Neerav Shah from the state of Maine CDC. Dr. Stray, thank you for your excellent presentation. I am trying to get a better understanding of the age dimension to this phenomenon. I noted on your slide 5 that among the AstraZeneca recipients, the median age was 40, which strikes me as roughly the median age of the background population. But among J&J recipients, the range was from teens to those in their 50s, suggesting a much lower median age. 2 questions. Is there a biological basis to believe there is an age component to this? And then secondly, to the extent there are differences with those 2 vaccines, could the lower age among the J&J recipients be merely a function of the way in which vaccines were rolled out in the U.S.? That is to say, older individuals perhaps received an mRNA vaccine earlier in the vaccination effort, and it's just now where younger folks are getting J&J vaccines.
So it's an excellent point. I'm not certain. I think we're still in the data acquisition phase, so we haven't had that many cases, and so these numbers may change. The numbers that at least those are the published cases on the AstraZeneca vaccine. They've had a lot more time to accumulate cases, and we're still very early in the process of examining J&J cases. I would also say that the higher age range that some of those older patients from the UK experience, they seem to be a little different than the patients that were reported by the German, Austrian group and the Swedish group, and so I just wary whether we may have accumulated a couple of patients that may actually not have had the syndrome, because there were some people in the UK experience that had pulmonary embolism and didn't have cerebral venous sinus thrombosis or the abdominal thrombotic events. And HIT testing is particularly the functional assays are very challenging, and so I think I can't confirm whether there's differences in the vaccines, and that's why the age range is a little older with the AstraZeneca versus the Johnson & Johnson. I think our data are too preliminary, and maybe Sarah or Tom will have more data than I have, because I think they do a closer analysis of the U.S. cases than I've done. I've done a very thumbnail sketch of them, so I don't want to mislead you.
Thank you, sir.
Yeah, sure.
And yes, Dr. Oliver will present more data on that. Dr. Lee, your hand is up again.
Yes, I have a basic question to ask, which is if you could explain to me the, perhaps why the functional assays in the U.S. cases were different from the European cases. Is that a matter of a different assay being performed? And if whether or not that's the case, I guess my question is, if the assay is negative, does that mean platelets are activated in that instance?
That's an excellent question, Dr. Lee. Functional HIT assays, I think, are a very challenging test to do, and so that's why you have very few labs doing functional HIT assays. I think there's a handful of labs in the U.S. that do the serotonin release assay, which is the functional assay that was used in the U.S. cases predominantly. And that assay, let's say the reliability of it, I think varies depending on the expertise of the lab doing the tests. In the European cases, they use several different assays. Andreas Greinacher used his homegrown platelet activation assay that he's been doing since 1990. And so he's literally the world's expert on doing that assay and has more experience with that assay than anybody else. And so I tend to place a lot of faith in his functional HIT testing. The UK paper used a flow assay, and there have not been lots of papers comparing these different functional assays and their reliability, their sensitivity and specificity, et cetera. So I think it's premature to say that the functional assays results we're getting in the U.S., that those patients didn't have platelet activation. I just worry about the reliability of the assay, because it's such a difficult test to do. And when you have a lab like Andreas Greinacher's getting it consistently, he's just such an expert. I think that I put more weight in his assay. So I think for our testing, as a clinician, what I'm going to be putting my weight on is, and my the emphasis on diagnosis is going to be the clinical situation, the clinical presentation of the patients, the thrombocytopenia, and then if they have a positive ELISA assay, which seems to be very sensitive for this, since I think all the ELISA tests have been positive in these patients, that's going to be what tells me this person has TTS, and we need to treat them like that. And if I get a negative functional assay back, I'm not going to stop treating them like they have TTS. At least that's my take right now. And this, of course, is, again, a moving target. It's going to change, I think, as we get more experience. Because we're only talking about 5 functional assays that have been done so far, I think, in the U.S. on our patients.
Thank you.
Are there any other questions? Dr. Shaw, your hand is up. Is that still a residual from your first? Thank you.
My fault.
No, it's fine. Anyone else? I don't want to close discussion before everyone's had a chance. Very good. Again, thank you very much for that presentation and for your answers to all the questions that we posed to you. Thank you again.
Thank you very much.
Our next speaker will be Dr. Shimabukuro, who will talk about thrombosis and thrombocytopenia syndrome, TTS, following Janssen COVID-19 vaccine. Dr. Shimabukuro, please.
Thank you, Dr. Romero. Next slide. Next slide. So I'll be covering some background, which will include just refamiliarization with some of the material that was covered at the last ACIP meeting and some of the data that led up to that meeting. And then I'm going to discuss thrombosis with thrombocytopenia syndrome, or TTS, following Janssen COVID-19 vaccine. And then I'll summarize my findings. Next slide. Next slide. So I mentioned thrombosis with thrombocytopenia syndrome. So I do want to cover some basics. And thrombosis occurs when blood clots block blood vessels. And thromboses can be venous or arterial. And common complications can include heart attack, stroke, and infarctions. Causes and risk factors include trauma, immobility, inherited disorders, certain autoimmune disease, obesity, hormone therapy or birth control pills, pregnancy, smoking, cancer, and old age, just older age, just to name some. Symptoms include pain and swelling in an extremity, chest pain, numbness or weakness on one side of the body, and sudden change in mental status. Thromboses are diagnosed primarily through imaging and blood tests. Next slide. So I mentioned thrombosis. Now I want to talk about platelets and thrombocytopenia. So platelets, which are also called thrombocytes, are colorless blood cells that help blood clot. A normal platelet count is 150,000 to 450,000 per microliter. And in the healthcare world, that's usually shorthanded 150 to 450. Platelets stop bleeding by clumping and forming plugs in blood vessel injuries. Thrombocytopenia is a condition in which you have a low platelet count, meaning below 150,000 per microliter. Dangerous internal bleeding can occur when your platelet count falls below 10,000. Though rare, severe thrombocytopenia can cause bleeding into the brain, which can be fatal. Next slide. So just to provide some context around the vaccine safety issue that we are discussing, this issue first came to attention because of the rare blood clots with low platelets that were detected following the AstraZeneca COVID-19 vaccine. And many of these events involved cerebral venous sinus thrombosis, which Dr. Strife described quite well in his excellent presentation. Next slide. So this is a screenshot of the presentation at the last ACIP meeting, where we focused on reports of cerebral venous sinus thrombosis with thrombocytopenia after the Janssen COVID-19 vaccine. Next slide. And I'm not going to cover this because Dr. Strife covered in detail the anatomy and the actual condition. Next slide. So this was some of the basic surveillance data that was presented at the last ACIP meeting on April 14th. And at that time, we had 6 reports of CVST with thrombocytopenia following 6.86 million doses administered of the Janssen COVID-19 vaccine. That translated to a reporting rate of 0.87 cases per million doses administered. In contrast, there were 0 reports following 97.9 million Pfizer-BioNTech doses administered, and 3 reports following 84.7 million Moderna COVID-19 vaccine doses administered. However, all those 3 reports had normal platelet counts, so this was essentially 0 reports following 180 million doses, just over 180 million doses of the mRNA vaccines, and we'd considered this a reporting rate imbalance. Next slide. So the take-home message at that time was that CVST is rare but clinically serious and can result in substantial morbidity and mortality. It's usually not associated with thrombocytopenia. We did an observed versus expected analysis, which was a bit of an apples and oranges comparison because our expected was based on CVST. We don't really have good information, as Dr. Streif pointed out, on the background incidence of CVST with thrombocytopenia. It's extremely rare. The reports were in women age 18 to 48, all with thrombocytopenia, so these were younger women, and there was no obvious patterns of risk factors detected. Again, by contrast, CVST with thrombocytopenia had not been observed after the 2 authorized mRNA vaccines. The clinical features of the Janssen cases were similar to those observed following the AstraZeneca COVID-19 vaccine in Europe, and both the Janssen and the AstraZeneca vaccines contain replication incompetent adenoviral vectors. Next slide. So based on the information, actually prior to the ACIP meeting, CDC issued a HAN, and the bottom line in that HAN was that CDC and FDA recommended a pause in the Janssen COVID-19 vaccine out of an abundance of caution while further assessment of the signal could occur. And an important message to healthcare providers was that they should be aware of these potential adverse events and so they can provide proper management due to the unique treatment requirement of this type of blood clot, which is not giving heparin if the HIT test is positive, treating with other non-heparin anticoagulants. Next slide. So after we detected this safety signal in our VAERS system, we turned to our vaccine safety data link system, our population-based system, to see what kind of data we had on this rare condition, CVST. And our initial focus was on looking at the Pfizer, BioNTech, and Moderna vaccines, and I will present data on the Janssen vaccines. But as of April 17th, there were 2.7 million doses of Pfizer, BioNTech, and 2.5 million doses of Moderna COVID-19 vaccines administered in VSD. So a substantial amount of the mRNA vaccines administered in the VSD. There were 10 total cases of CVST identified following the mRNA vaccines. 5 of these cases ruled out. They were either historical cases or had a history of head injury, and 1 had a history of chronic cavernous sinus syndrome. There were 5 cases that were potentially CVST, but all without thrombocytopenia. So after this initial the supplementary VSD analysis for mRNA vaccines, there were no confirmed cases after 5.2 million doses of mRNA COVID-19 vaccines administered in the VSD. Next slide. So our assessment at that time is that we had a safety signal detected for CVST with thrombocytopenia following the Janssen COVID-19 vaccine. 6 cases observed in women 18 to 48 years in early post-authorization monitoring. I'll mention there was 1 case observed in the pre-authorization clinical trials in a 25-year-old male. And currently, there is a lack of evidence of an association between mRNA COVID-19 vaccines and CVST with thrombocytopenia. Next slide. I want to mention that the Brighton collaboration has a draft case-finding definition for a condition which is called thrombosis with thrombocytopenia syndrome, which is broader than just CVST with thrombocytopenia, includes other clots. Hallmark of their draft case definition is that you need to be thrombocytopenic, so platelet count less than 150. And in addition to rare thromboses, it includes more common thromboses such as deep vein thrombosis, pulmonary thromboembolism, ischemic stroke, and myocardial infarction. Next slide. Now I'll move on to the data sources and the TTS cases that we have detected and assessed. Next slide. Our main source of cases is the Vaccine Adverse Event Reporting System, which is the nation's early warning system for vaccine safety. It is a spontaneous reporting system or a passive surveillance system that's co-administered by CDC and FDA. VAERS accepts reports from anyone, so patients, parents, caregivers, healthcare providers, and vaccine manufacturers. As a national system, it has a large population potentially under surveillance. It's good at detecting rare adverse events and rapidly detecting safety signals. It's subject to the limitations of passive surveillance in general, and it is not designed to assess causality. Next slide. I want to mention that in addition to our VAERS system and our VAERS team of reviewers and abstractors, we also relied on our clinical immunization safety assessment project team and our consultants at our CISA sites to help review and evaluate these cases. Next slide. So case finding for TTS following Janssen COVID-19 vaccine occurs in a variety of ways. Often healthcare providers will directly contact CDC with potential TTS cases as they come into the hospital or as they're hospitalized. CDC initiates an investigation and facilitates submission of a VAERS report if it hasn't already been reported to VAERS. Also, FDA physicians review incoming VAERS reports daily to identify potential TTS cases. We also search the VAERS database for possible TTS reports. And this includes MEDRA PTs, a search for MEDRA, that's the medical dictionary for regulatory activities, which is a standardized coding that's used for regulatory purposes. We do a MEDRA PT preferred term search for large vessel thrombosis and or embolism, any report. We don't restrict it to reports that have MEDRA PTs for thrombocytopenia or any indication of thrombocytopenia in the narrative. For this particular analysis, we did not include the more common thrombosis events that I mentioned in the Brighton collaboration TTS case definition. These events will be evaluated in a subsequent analysis. Medical records are requested for all potential TTS cases to confirm thrombosis with laboratory evidence of thrombocytopenia. And CDC and FDA medical officers reviewed TTS reports and available medical records, and CISA experts, including hematologists, were consulted as needed. Next slide. So here is really the high-level bottom line for reporting rates of TTS after Janssen COVID-19 vaccine. So as of April 21, we had almost 8 million vaccine doses administered with 15 confirmed TTS cases. Some of this age and sex-specific doses administered data were imputed because there were missing variables for age or sex. That was a small minority of the data, though. There are additional potential TTS cases under review, including some potential male cases. I do want to mention that one case of TTS was excluded from the final analysis. This was a female less than 50 who had concurrent diagnosis of COVID-19 and had a very complicated clinical picture. We weren't comfortable including that as a case, but we wanted to mention that for transparency. So in this table, on the left-hand side there, you have the age groups, which we have split out 18 to 49 years old and 50 plus. Then you have the female data on the females and data on the males. All these 15 cases were in females, and 13 of the 15 were in the 18 to 49-year-old age group, the remaining 250 plus. And you can see the doses administered there in the next column, and that translates into a reporting rate of 7 TTS cases per million doses administered of the Janssen COVID-19 vaccine for women aged 18 to 49 years old. For women 50 plus, this translates to 0.9 TTS cases per million doses administered. You'll see on the right-hand side of this table, there were 0 cases in males in either of these age groups, so the reporting rate is 0 per million. That does not mean there is no risk in males. There could be cases that we did not identify in the database. Theirs is also subject to underreporting, so there could be cases that simply weren't reported. And there could be cases that become apparent later on that just have not appeared. But as of now, we had 0 cases in males, and thus the reporting rates of 0 per million for both age groups. Next slide. When we looked at the graph of patient age in years, you can see here's the interval from 18, or it's the complete interval, 18 plus. But you can see this apparent clustering of individuals in the mid to late 30s. When we observed this clustering, we decided that it would be prudent in consultation with our vast colleagues and the work group. It would be prudent to take a closer look, splitting the age intervals a little bit finer in women. So if you could go to the next slide, please. So this is that analysis where we're taking the female recipients of COVID-19 disease and the cases and basically splitting them into finer age groups. For this analysis, we had nearly 4 million doses administered to women, and again, all 15 confirmed cases were in women. If you look at this, you focus in on the 30 to 39-year-old age group. This is where we saw the most cases, and this is where we, based on doses administered, had the highest reporting rate, which was 11.8 cases per million doses administered. And you'll see in the older age groups, 50 to 64 and 65 plus, the reporting rates are lower than in these younger age groups. Next slide. So some of the characteristics of these case patients: the median age was 37 years, range 18 to 59. Median time to symptom onset was 8 days, range 6 to 15 days. Again, all cases in females. 12 of these cases were cerebral venous sinus thrombosis cases. There were no cases that were pregnant or postpartum. There were 2 cases with COVID-19 disease. However, these were both by history and fairly remote, with no documentation of serology testing. As far as some of these identified risk factors, 2 case patients were using oral contraceptives. Obesity was fairly common. There were no patients with known coagulation disorders. Next slide. So here's a graph of time from vaccination to symptom onset, range of 6 to 15 days. I think the take-home message from this is these cases first experience symptoms usually around 1 to 2 weeks between the first and second week post-vaccination. Next slide. So this slide shows the signs and symptoms in patients with cerebral venous sinus thrombosis after Janssen COVID-19 vaccine. It's just limited to those with CVST, a subset of the TTS cases with CVST, which is 12. The initial presenting symptoms, and this was restricted to individuals, or at least 2 patients had experienced these symptoms. Initially were headache, all of which started 6 or more days after vaccination: chills, fever, nausea, vomiting, malaise, lethargy, and abdominal pain. And you can see some of the signs and symptoms later in the clinical course. I think the important message on this slide is that these initial symptoms are fairly vague and nonspecific, mainly headache. But importantly, the headaches for these started 6 or more days after vaccination. I say that because headache is a commonly reported adverse reaction after vaccination, particularly after the COVID vaccines. But for the reactogenic headache, that usually occurs on the day after vaccination, and not starting 6 or more days after vaccination. So that's unusual. Next slide. So the locations of thromboses in these TTP patients, and these are not mutually exclusive. You see the locations for CVST there, transverse sinus, sigmoid sinus, confluence of sinuses, straight sinus, superior sagittal sinus, inferior sagittal sinus, and cortical veins. And then for the other locations, portal vein, hepatic vein, superior mesenteric artery, splenic artery, pulmonary artery, and other vessels, as you see there. 7 patients with CVST did experience an intracerebral hemorrhage. Next slide. So these are some of the selected laboratory findings for these TTS patients. All were thrombocytopenic. That's really a part of the case definition. 10 were severely thrombocytopenic, had platelet counts of less than 50, 3 between 50 and 100,000, and 2 between 100,000 and 149,000. When testing was done, all of the cases had a platelet factor 4 heparin-induced thrombocytopenia positive ELISA antibody test, and 4 testing was either not available or wasn't done. Next slide. Here's some SARS-CoV-2 testing results. I think the message here was fairly unremarkable, although I will mention we did have that one potential case which we excluded, and that was based on the determination that was somewhat of a complex and unique case, and I can provide more information on that if the committee wishes. Next slide. These are the treatment and outcomes among the TTS patients. 6 received heparin, 12 got non-heparin anticoagulants, 7 platelet transfusions, and 8 got IVIG. All those individuals that got treated with heparin, all 6 of those individuals, that happened before the HAN was released. So I think that's at least some indirect evidence that healthcare providers got the message in the HAN not to treat with heparin unless the HIT testing was negative. And then outcomes, there were 3 deaths, 7 remain hospitalized, 4 in intensive care, and 5 have been discharged home. I'll mention that in those 3 deaths, none of those patients received heparin. Next slide. Now I want to cover some additional analysis we've done in our vaccine safety data link. Just to remind you, that is our population-based system that we use for near real-time surveillance and research. It has data on over 12 million persons per year, mainly from electronic health records and administrative data. Next slide. So I apologize, this is a busy slide, but I felt it was important to fit it all in on one slide, and I'll walk you through it here. This is VSD data on thrombosis events after Janssen COVID-19 vaccine. So I'll just mention there were 142,000 Janssen COVID-19 vaccine doses administered in VSD through April 17th. There have been no statistical signals detected for any pre-specified RCA outcomes. Of course, there's a relatively small number of doses, at least compared to the mRNA vaccines. There have been no CVST cases identified following the Janssen COVID-19 vaccine. There were 22 VTE and or PE cases identified in the 1 to 42 days following vaccination, which were quick reviewed. And these aren't mutually exclusive, so this includes 2 with both VTE and PE. So of these 22 cases, 6 ruled out as not VTE. 16 were confirmed as VTE and or PE. 4 had symptom onset prior to vaccination, and that includes 1 case with thrombocytopenia documented prior to vaccination. 1 had an indeterminate onset, and that left us with 11 incident cases following vaccination. Of these 11, 6 were female. That was 2 PEs and 4 VTEs. 5 were male. That's 1 PE, 4 VTEs. The ages ranged from 50 to 79 years. None with a history of COVID-19 infection, and none with thrombocytopenia at the time of VTE or PE. So, and I'll say we're using a bit of a broader case definition than we used for the VAERS analysis. We're including some of these more common thromboses for for the Janssen VSD assessment here. But the bottom line is we have a relatively small number of Janssen vaccine doses in VSD, but we did not see any potential cases of thrombosis with thrombocytopenia based on this analysis. Next slide. Next slide. So to sum up, TTS is rare but clinically serious and potentially life-threatening, and a potentially life-threatening adverse event that has been observed in association with the Janssen COVID-19 vaccine. Symptom onset appears to occur at least several days after vaccination, typically around 1 to 2 weeks after vaccination. The clinical features of TTS following Janssen COVID-19 vaccine appear similar to what is being observed following the AstraZeneca COVID-19 vaccine in Europe. It is important to recognize TTS early and initiate appropriate treatment. Do not treat TTS with heparin unless HIT testing is negative. The U.S. Vaccine Safety Monitoring System is able to rapidly detect rare adverse events following immunization and to quickly assess safety signals. Safety surveillance and research on TTS continues, and CDC is committed to open and transparent communication of vaccine safety information. Next slide. So as far as next steps, we'll continue enhanced monitoring in VAERS and continue surveillance in other vaccine safety systems to include VSD, the CMS data, and the VA electronic health record. That's with our colleagues in FDA and the VA. We plan to expand the VAERS database search strategy for TTS reports. This would include the MEDRA preferred terms for large vessel thrombosis and thromboembolism. Again, all reports, regardless of the presence of thrombocytopenia, which we go in and review to confirm thrombocytopenia, but also MEDRA PTs for more common thrombotic events and MEDRA PTs for thrombocytopenia or text string searches for thrombocytopenia or low platelets in the narrative section of the VAERS reports. So that's using a combination. You have to have one of these more common thrombotic events and also evidence in the report of thrombocytopenia. And we will review the medical records for all potential TTS case reports to confirm the thrombosis and to also confirm the presence of thrombocytopenia. Next slide. And just to remind healthcare providers, VAERS is our system for collecting rare, serious adverse events. Anyone can report to VAERS. You can go to the VAERS website and submit a report on the online portal. There are some resources there for assistance if you need help with the system. And just to remind healthcare providers and our public health partners that if we do request medical records, we appreciate cooperation, and HIPAA permits reporting of protected health information to public health authorities, including CDC and FDA, when they're conducting these activities as a public health function. Next slide. I wish to acknowledge the contributions of the investigators from these following organizations. Next slide. I'll be happy to take your questions.
Thank you again, Dr. Shimabukuro, for another excellent and well-presented review. So we're going to open this up to questions. Let me explain that we will go until about 5 minutes before the hour, then take a 5-minute break, and then begin the public comment session. If there are additional questions or further discussion necessary, I will return to this topic after the public comment session. So let me begin with the first hand. It was Dr. Paling. Dr. Paling, please.
Thank you. Dr. Shimabukuro, thank you for an excellent presentation. I think this data clearly demonstrates that the safety systems that have been set up with the FDA and the CDC are working in detecting the events we need to see and providing us much needed information. As I understand the presentation from the VAERS, there were 15 confirmed cases, and I believe the slides indicated there are potential cases of TTS under review. My first question is, could you share how many of those cases are in the potential review, under review status? And then the second is, could you share information on the case that was excluded? Thank you.
Sure. I'll say it's a relatively small number of cases that are under review. That's kind of a dynamic number, but it's just under 10 right now. But that does change on a daily basis. That's the short answer to the question, and that's when we're looking at our relatively restricted case definition, where we're looking at large vessel thrombosis in these unusual locations in the presence of thrombocytopenia. I had mentioned that we are going to expand our case definition to make it more consistent with the draft Brighton collaboration case definition. That includes other thrombosis events like venothromboembolism, DVT, PE, ischemic stroke, acute myocardial infarction. When we do expand beyond that, I anticipate we'll have a substantially larger number of cases under investigation. That's normal. Those events are pretty common and commonly reported to VAERS. So I would say using the more restricted case definition, it's a relatively small number, but that number could expand substantially when we broaden out our case definition. Oh, and let me go to, if you could advance the slide one more. So here's information on the report of TTS that was excluded from the case count. It was in a female, less than 50, that had COVID-19. She was PCR positive and had a complex clinical course. She was hospitalized twice, admitted first time 22 days after vaccination with COVID-19 pneumonia, and she presented with nausea, hematemesis, shortness of breath. The date of symptom onset was not clear, and at that time she had a normal platelet count. She was readmitted 28 days after vaccination, which she presented with nausea, hematemesis, abdominal pain, shortness of breath, and cough. That time she had a platelet count of 100,000, and the imaging studies showed CVST and DVTs and a PE, and she subsequently died during hospitalization. The reported cause of death was respiratory failure, shock, and COVID-19 pneumonia. So as you can see here, this was a complex clinical course where the cause of, it was just not so clear whether this should be included as a case or not. We ultimately decided in consultation with our colleagues at FDA that we would not include this in the final analysis, but we would certainly mention that this case existed and for transparency have this information available for the committee.
Thank you. Dr. Bernstein?
Yeah, great presentation as always, Tom. You mentioned in your summary that TTS after the J&J vaccine is similar to the AstraZeneca experience. So it seems that viral vector vaccines are associated with TTS. Your latest data suggests 15 cases per 4 million doses of J&J in the United States. Can you remind me what the incidence was in AstraZeneca's in Europe and the UK?
I don't have that information at hand. I would have to get back to you on that, Dr. Bernstein, unless somebody else on the call has better insight on the AstraZeneca data.
Dr. Shimabukuro and Dr. Bernstein, we have that laid out in the in my presentation coming up. So I'll let you defer to that, and we'll walk it out. If you have questions, Dr. Bernstein, after that, I'm happy to answer.
Great. Thank you.
Thank you very much. Ms. McNally.
Thank you. I think this question is for Dr. Shimabukuro, but it might be more appropriate for Dr. Mishler. My question is that, well, obviously this is a rare outcome. It is potentially life-threatening. And I'm mindful that the compensation for injury is under the CICP, the Countermeasures Injury Compensation Program. And I'm wondering if a vaccine injury table is being developed for the COVID-19 vaccine. Thank you.
This is Tom. I'm going to have to defer to my colleagues at HRSA on that one.
This is Captain Mishler. The vaccine injury table will be developed, but it is not presently under development. We will have injuries that are on the table, but we also give equal consideration to non-table injuries as well.
Thank you. Dr. Long.
Yes, thank you so much. First is a comment. The Brighton definition uses a very liberal definition of thrombocytopenia, and I think that it's very likely that that will become contaminated, or you'll have to ratchet down an awful lot till you get to this syndrome, which is by definition, it's an autoimmune type syndrome, which is characterized by extremely low platelets in many, many patients. So I don't know how they chose. I think it was 150,000. You're going to have a lot of things that aren't it. So we're glad that today you've presented us with the cerebral venous thrombosis part, sinus part. My question is, do you have data for the time between onset of symptoms, especially headache, and first coming to medical attention, so we can understand a little bit better if this is rapidly progressive and potentially fatal before any medical intervention could occur, or if there are 4 or 5 or 6 days in which we could educate and potentially save lives? Thank you.
I think your question is time to admission from vaccination. So the person gets vaccinated, and when are they admitted to an acute care facility? Is that what you're looking for?
Not since vaccination. I'm thinking since onset. You had a column of initial symptoms. And I also think that might be time-wise a little contaminated by fever and things that could have been still reactogenic kind of things. But the headache that has the onset at 6 days and beyond, what, for instance, is the time period before that and coming to medical attention? Or we would take hospitalization as a possible marker, although they could have been sent home from emergency rooms if we had nonspecific headache.
I'm kind of scanning my additional documents here. I think what you're asking for is the median time and the range from symptom onset to when they are actually hospitalized. That when they have symptoms and when somebody recognizes that this is a problem and they admit them to the hospital. The median time for that is 5 days, and the range is 0 to 14.
Thank you.
Dr. Dushan.
Thank you. Thank you for this presentation, Dr. Shimabukuro. My question is about the next steps. what is the plan for updating available information, and how will that be made available to the public?
Well, we will certainly continue our enhanced surveillance for this condition, our case finding, our rapid assessment of cases, our requests for medical records, our review, our consultation with our CISA colleagues to do a deep dive into these cases. We have added CVST to our VSD rapid cycle analysis, and we are looking into ways where we can automate and become more efficient for detecting thrombosis with thrombocytopenia in our population-based systems, and we will continue to brief the technical subgroup on a routine basis. They meet weekly, and we're happy to brief the work group at request, and we will certainly come back to the full ACIP whenever we are asked, and we'll try to publish this information as soon as possible. We have publications in the works right now, but certainly we are happy to come back to the ACIP and present safety data whenever that's needed.
I thank you very much. I mean, this level of scrutiny really leaves no doubt that vaccine safety is a top priority for the CDC, for the FDA, and our national program. And I know the public will be interested in hearing ongoing updates of what you're finding. So thank you.
Thank you. I see no other hands. Dr. Daly, sorry, forgive me. Yes, Dr. Daly.
No, it's okay. I just put my hand up. So I would just echo the comments of Dr. Duchin and Dr. Paling, just that the very fact that we can detect something this rare is an indication of the strength of the vaccine safety monitoring system, and that monitoring is going to continue. So that's the comment. And then the question, Tom, is about the benefits of the pause. Is it your sense that sort of, at least by my count, the pause has been on for 11 days, and is it your sense that the pause achieved the goal of better understanding these cases and better characterizing them?
We would have done this work regardless of whether there was a pause or not. I mean, it's certainly the entire totality of the situation really caused us to focus in on rapidly assessing these cases. We changed our procedures. We staffed up to do this. So I mean, I guess you could argue that the act of the pause did really focus our efforts on really getting to the bottom of this quickly, so we could come back to the ACIP 10 days later.
This is Melinda Wharton. Can I make a comment?
Of course, Dr. Wharton, please.
I think there was a very important objective achieved by the pause, which is allowing clinicians to be informed, about the condition, raising public awareness, and providing time to get a better assessment of risk so that we can have the conversation we're having today.
Thank you for those comments. Dr. Fink, please.
Thank you. I just want to mention on behalf of FDA that we agree with the statements by Dr. Wharton and Dr. Shimabukuro with regard to the benefit of the pause. And in terms of ongoing efforts, of course, FDA will continue to work closely with CDC on evaluating and characterizing these cases and this adverse event. And if any further information comes to light that would warrant additional safety communications or even changes to the warning statement to ensure adequate risk mitigation, then we would, of course, do that. Thank you.
Thank you. Any other comments? I'm looking around my room to see if anybody from the liaisons or from the Voting members,
filed a lawsuit against HHS for not conducting any vaccine studies in over 30 years, which I cannot believe. In 1986, Ronald Reagan took all liability away from any vaccine manufacturer, and it blows my mind that the vaccine manufacturers cannot be held accountable for any of the deaths caused from the vaccines that they create. My wife, Priscilla, is a first responder in Millersville, Georgia, and the amount of calls that come through the dispatch office pertaining to shortness of breath or non-responsive following the COVID vaccine is unreal. I also have a childhood friend that works in the dispatch office in our area, and they aren't even allowed to talk about the vast amount of calls that come through following the COVID vaccine. I mean, it doesn't make any sense why nobody can talk about what's going on in the real world versus these clinical studies that are so-called being done. Since we have stopped vaccinating our children, they are healthier and happier. I love my family and my children, and I'll do anything to protect my family. And from what I have seen and encountered with the vaccine, they don't work for my family. So my wife and I have our story on YouTube under Will and Priscilla Zelinsky that pertain to all the details following our child's vaccine at 4.5 months old. But the main focus needs to go to all the deaths that are deriving from the COVID vaccine, which I haven't seen any good clinical studies pertaining to how they are healthier and they're working. But I appreciate your time, and I thank y'all for allowing me to speak. Thank you.
thank you for your comments. Mr. Thompson.
Hi, good afternoon, everyone. My name is Joel Thompson, and I come to you today as a concerned member of the American public. To the ACIP members present today, I first want to commend you on the tremendous thoughtfulness, thoroughness, and care you have repeatedly demonstrated in considering COVID-19 vaccines. The American public can take comfort knowing dedicated public servants such as yourselves are continually examining the COVID-19 vaccine. Unfortunately, however, with regards to the Janssen COVID-19 vaccine, my compliments end here. First and foremost, I am dismayed by the paternalistic attitude displayed by members of ACIP towards the American public. Your concerns noted over the last week towards Janssen's COVID-19 vaccine would be an appropriate basis to withhold vaccination if individuals were being compelled or coerced into receiving the Janssen vaccine. That is not the case. Every person who is receiving the vaccine is doing so consensually. Your actions, or rather lack of actions a week ago, have taken agency away from individuals to make their own personal health decisions about what they feel is best for their own individual situations. They are now not able to choose to take the Janssen vaccine. The overwhelming recommendation of public health officials is to take the vaccine you are able to first. While the first vaccine I was able to receive was Moderna's, if the first vaccine I had been able to get were Janssen's, I would have gladly taken that, even knowing what we all know today. And furthermore, I would have been thoroughly dismayed if I had had to delay vaccination because I had originally been scheduled to receive Janssen. Second, regardless of my concern about paternalism, it has been clear since before last week's ACIP meeting that the benefits of the Janssen COVID-19 vaccine vastly outweigh the risks, and especially in hard-hit areas that will benefit from the vaccine's single-dose regimen, such as the frightening surge we are seeing in Michigan. Don't just take my word for it. For example, last week, Dr. Jeremy Samuel Faust of Brigham and Women's Hospital, an instructor at Harvard Medical School, and Dr. Ashish Jha, dean of the Brown University School of Public Health, both posted threads on Twitter explaining why they believe the decision to continue the pause was a mistake, because the benefits of the Janssen COVID-19 vaccine clearly outweigh the risks in the current environment we are in today. There is only a single responsible path for ACIP to take today. That path is to correct last week's mistake and let Americans make their own decisions about their bodies and their own health care, including a decision about whether or not to take the Janssen COVID-19 vaccine. Thank you very much for letting me speak to you today.
Thank you for those comments. Mr. Ferrugia.
Hello, I'm Brett Ferrugia. I'm representing myself as a private citizen today. Since the pause on Johnson & Johnson vaccines nearly 2 weeks ago, vaccination rates across the nation have fallen by 13%. In more hesitant states, vaccination rates have fallen further, including 33% in Texas and nearly 50% in Wyoming. This cannot fully be attributed to a reduction in supply due to the pause, and across several states, there have been mass cancellations of all 3 manufactured vaccines. In addition, including in my state of New York, homebound programs were halted for over a week due to the pause. Today, I urge the committee not only to immediately reinstate the Johnson & Johnson vaccine in order to reassure the public, but also make efforts to repair the severe damage done to vaccine confidence across the nation by this overzealous pause. I also urge the committee to reevaluate risk assessment practices. It is a near statistical certainty that more people will die of COVID-19 due to 10 million doses of vaccine sitting idle at vaccination sites across the nation, combined with renewed skepticism induced by the pause, than would die from exceedingly small chances of blood clots. Further, during the pandemic, the ACIP's decisions have had much more far-reaching effects than just vaccine safety. When the pace of vaccination is both life and death for people and livelihoods, the committee should weigh the communal good against individual safety. I also urge the committee to take into account how many people will never get vaccinated due to the pause of this vaccine and weigh it against the exceedingly rare chance of death from blood clots. This pause may end up being the worst blow to vaccine confidence since the fraudulent link between vaccines and autism was made 23 years ago. If we still have this falsehood about autism and vaccines spreading 23 years later, how long will it take to quell the myth that vaccines cause blood clots in any statistically meaningful way? Again, I urge the committee to immediately reinstate the Johnson & Johnson vaccine with the necessary warnings to boost public confidence, increase vaccine supply, and allow homebound and underserved communities access to a one-dose vaccine. Further, I urge the committee to reevaluate risk assessment practices to better reflect the risk-benefit balance during the unique times of a pandemic. Thank you for your time.
Thank you for your comments. Ms. Berry.
Hello, can you hear me?
Yes, we can.
Awesome, thank you. Hello, my name is Sarah Berry, and I am an independent pro-vaccine advocate who operates online under the username 42Believer. I was incredibly grateful to share my public comment with you last time, and I am equally excited to do so again. Over a month ago, I received the Johnson & Johnson vaccine and experienced no other side effects other than a sore arm and mild flu symptoms. I was and still am very grateful to have received this vaccine, and I am grateful to the ACIP for continuing to monitor the situation and for having these transparent discussions. Last time I gave comment, I ended with a strong phrase that was anti-vaxxers abuse autistic children. I am often asked what I mean by this, so I would like to take the time to educate you all on this lesser known but crucial aspect of the anti-vaccine community, as I truly believe that highlighting these transgressions is the only way to win against them. When I have discussions with people, whether they think that they're super pro-vaccine or whether they're on the fence or not, most of them have heard of people like Andrew Wakefield, but they really don't know about Andrew Wakefield and the things that he's done beyond getting his license taken away and beyond having the study that he published be retracted. When I hear Andrew Wakefield, the first name that I think of is Jack Piper, because he was one of the 12 children that was part of the original study, the original Lancet study. And Jack Piper's family was able to get hundreds of thousands of pounds in compensation from the hospital because he suffered intense cuts to his interior colon as a result of the procedures that were performed on him during the study. This is the first example of the modern anti-vaccine movement using children as autistic children in particular as experiments to try and cure them. Another person that most people, pro-vaccine or not, most people haven't heard of people like Mark Geyer. Much like Andrew Wakefield, Mark Geyer lost his medical license, but instead of the experiments that Wakefield did, he gave autistic children a drug called Lupron, which ended up chemically castrating autistic children. And he charged parents $12,000 a month for the privilege of experimenting on their children without informed consent. There's a lot of anti-vaxxers who talk about we need informed consent and we need everything to go through the proper process and be tested against placebos. And I genuinely want them to look in their own communities and see that their own communities endorse and enable people like Mark Geyer to continue to hurt autistic children and flout the very standards that they are trying to hold big pharmaceutical companies to. And again, I strongly feel that this particular argument is the only way to win against people who are on the fence.
Thank you for your comment. Your time has expired.
Thank you very much.
Next speaker is Mr. Cambiero.
Yes, hi. I'm Juan Cambiero. I'm in the biology department at Hunter College. I do COVID forecasting work for Metaculus. And most importantly, I'm a concerned member of the public who fears that this pause is having a very negative impact on getting life-saving vaccines into as many arms as possible. I would like to strongly urge the committee to recommend lifting the pause on the J&J vaccine and to recommend rollout be resumed with no age or sex restrictions. There are 4 key reasons that support this. First, the risk-benefit profile of the vaccine is very favorable, even for young people. As was presented by Dr. Shimabukuro earlier, data that has come in since the pause has confirmed that TTS is an extremely rare event, with a risk somewhere in the range of less than 1 to 10 in a million. Positive risk-benefit profile has also recently been affirmed by the European Medicines Agency on April 20th. And this positive risk-benefit profile is clear even for young people for whom the risks of COVID are clearly far greater than the risks from this vaccine, including, as Dr. Streif mentioned, even the risk of TTS, which is somewhere around 10 times higher from COVID than from the J&J vaccine. Current polling indicates that there is a significant group of young people, including young women, who are somewhat vaccine hesitant and who favor the fact that this vaccine is one and done. For this group, resuming J&J vaccine rollout is crucial since these people might otherwise not get vaccinated. Second, as was pointed out by Dr. Cotton at the last meeting, the J&J vaccines lack storage requirements, and the fact that its single dose means it's by far the best vaccine to vaccinate disadvantaged groups like the homebound and homeless. Fully vaccinating these vulnerable populations with 2-dose mRNA vaccines that require cold chain is in many cases not possible. I urge the committee to take into account these equity considerations. Third, although vaccine hesitancy in the general population doesn't seem to have increased as a result of this pause, polling by YouGov shows that the public's perceived safety of the J&J vaccine specifically has plummeted by 15%. Moreover, a Harris poll out this morning finds that among the wait-and-see group of Americans, 67% say that the pause has negatively impacted their impression of the J&J vaccine safety, and 62% say that it has negatively impacted their confidence in vaccine safety overall. An extension of the pause seems likely to dent public confidence even more, especially among this crucial wait-and-see group. Finally, although ACIP is responsible for making recommendations for the U.S., I urge the committee to also keep the global context in mind. Many countries take cues from what the U.S. does, have also suspended rollout of the J&J vaccine, even though they don't have access to other vaccines yet. Dr. Lee made this point at the last meeting, and I thought was worth re-emphasizing since it is so important. The longer everyone else remains unvaccinated, the longer this tragedy of a pandemic will go on, people will die, and new variants will arise. A lot hinges on your vote today, and I really hope you consider lifting the pause instead of continuing to restrict use of this life-saving vaccine. Thank you so much for your work and for considering my comment.
Thank you for your comments. Dr. Ratsan.
Thank you for the opportunity to speak at the ACIP today. My name is Scott Ratsan. I'm a public health physician, professor, and editor of the Journal of Health Communication. I teach at CUNY Graduate School of Public Health and Health Policy, New York, and serve on the National Academy's Board on Global Health. Previously, I was a member of the Board of Scientific Counselors for the CDC's Office of Infectious Disease. Last year, along with Dr. Heidi Larson and others, I helped found a global initiative called CONVINCE, which stands for COVID New Vaccine Information, Communication, and Engagement. Since last March, CUNY and CONVINCE have conducted more than 20 COVID-related consumer research studies in the U.S. and internationally. Earlier this year, I shared some of these learnings from this work with the HHS National Vaccine Advisory Subcommittee on Vaccine Confidence and made a number of recommendations to help build public confidence in vaccines. I speak today to you with hope and concern. Hope that we can get this vaccine back in the pipeline and hope that we can rebuild confidence in it. Concern, though, that a key element of our current approach may not be sufficient to attain the levels of confidence and trust we need to reach the herd immunity and end this pandemic. A study we did just last week nationally confirms that a substantial percentage, 78% of the American public, believes vaccination is the only way to end the pandemic. At the same time, our study and others suggest that at least 1 in 5 Americans do not believe that COVID-19 is that big of a risk and are not likely to receive this or any other vaccine. As we cross the threshold of a 50% vaccination rate in this country, our efforts to control COVID-19 are clearly at a tipping point. Demand for COVID-19 vaccines has fallen significantly in the past weeks, and many county health departments are shuttering their mass vaccination sites. Vaccinating the next 25% of Americans will require new strategies and better systems. I believe the benefit-risk data you're considering today will support returning the single-shot Janssen vaccine to the pipeline with better surveillance. However, I do not believe that the current surveillance system provides data that are strong enough, prompt enough, or convincing enough to give unvaccinated people the confidence they need to roll up their sleeves. Our current VAERS system is a passive one. It identifies vaccine injuries based on people's reporting adverse events to the local, state, or tribal health authority. We need a more active approach. I strongly recommend that the ACIP consider supporting the development at the federal and state level of a new unified system for real-time collection of data, a registry of everyone who gets a COVID-19 vaccine, which vaccine they received, and when they got it. A proactive registry will greatly strengthen what the FDA calls post-marketing surveillance by generating better and prompter data on the safety and efficacy of this and other vaccines. This is data that can be reported to Congress and the public to build confidence in the safety of COVID-19 immunization and reinforce emerging statistical evidence on the positive effect of vaccination. Finally, ACIP's actions today will have national and global impact. The steps you recommend should bring this enormously useful single-dose vaccine back into the global pipeline. You can all help build a more vaccine-literate and vaccine-supportive America.
Thank you for your comment. Your time has expired. Thank you. So that ends our public comment session for today. We are going to take a small break here until 10 minutes to the hour, and we'll resume at that point with a presentation from Janssen. So we'll see each other, or hear each other back at 10 minutes to the hour. Thank you. All right, welcome back from your break. We're going to move on to our next presentation, which is an update on Janssen COVID-19 vaccine by Drs. Mammon and Waldenstreicher. And please forgive me if I mispronounced your names. Are you ready to proceed?
We are. Yes.
Thank you very much. Please go forward.
So thanks very much to all of you for the committee for the opportunity to meet with you today. My name's Matai Mammon. I'm the global head of R&D for Janssen. We believe the Janssen COVID-19 vaccine, which I'll refer to as the J&J vaccine, has important and even unique benefits in our global effort to end COVID-19. In the weeks since our vaccine was authorized for use in the United States, 7.9 million people have received the J&J COVID-19 vaccine. During that time, we've seen this vaccine is not only vital and important option across a wide range of people, but that it may be uniquely good and in some cases really the only practical option for some people. For example, for those who face barriers to healthcare services. The vaccine's proven highly protective single-dose efficacy also against concerning variants of SARS-CoV-2 that are emerging as viral replication and selective pressures continue at a global scale, and that could benefit us all. And as a real credit to the US and European and our own safety surveillance systems, we've worked in close collaboration with the US, Canadian, and European health authorities to identify a very rare but very serious side effect of thrombosis with thrombocytopenia that appears at a higher rate than the background rate in the population. We recognize that this is a very rare but very important and serious risk, and Janssen fully supports awareness, education, labeling. With these efforts, our goal is earlier recognition, making this event more identifiable, diagnosable, treatable, and with improved chances of successful outcomes. Our priority since the discovery of these events has been to work very closely with health authorities to increase awareness among the general public and healthcare professionals. We fully support the important information addressing this event in the vaccine fact sheet and prescribing information. I'll begin today with a few relevant features of the current COVID-19 pandemic and move into a description of the benefits of the J&J vaccine. I'll highlight efficacy and other key attributes relevant to the current COVID environment. Then my colleague Joanne Waldstreicher, our Chief Medical Officer, will take you through the safety of the vaccine with a focus on the characterization of this very rare but serious event of thrombosis with thrombocytopenia, then conclude with our framework for how we think of harnessing the benefits of this vaccine for the US and the globe while responsibly managing its risks. Just this past week, there have been nearly 40,000 hospitalizations and over 4,700 deaths in the United States, and around 10x or more of that number globally. The pandemic continues to rage in the face of the vaccine rollout. Importantly, many people face obstacles to accessing a vaccine, and we know that the J&J vaccine has been particularly important to people who live in rural or remote areas, for example, and who lack the type of healthcare infrastructure required for other options. I want to review a particularly important feature of the J&J COVID-19 vaccine measured in our phase 3 trial. We demonstrated strong protection offered after severe disease after day 28 of a single shot, with 100% protection against hospitalization and 100% protection against death. And what's most notable is that this pattern of protection was true also in regions such as South Africa and Brazil, where the 484K containing mutations are dominant. In South Africa, for example, we saw equal efficacy to that measured globally, including 85% protection against severe COVID, 100% protection against hospitalizations, and 100% protection against death. These results are supported from an ongoing study in South Africa, where we have now over 280,000 healthcare workers vaccinated, and these healthcare workers have significant and sometimes repeated exposure to B1351, the South African variant. Of note, the participants are primarily women and primarily under the age of 50. As with our phase 3 study, there have been no hospitalizations or deaths after day 28, and only 3 breakthroughs after day 28 of the 280,000 vaccinated people. The B1351 variant contains 3 mutations and 1 deletion on the tip of the spike protein, rendering the neutralizing antibodies induced by all the vaccines about 80 to 90% less effective than they are against neutralizing the original strain of the virus. This goes for our vaccine too. We believe that the excellent efficacy we nevertheless maintain against the variants is in part due to the T-cell immunity that recognizes so many epitopes of the spike protein that's very challenging for the virus to circumvent the vaccine. And our vaccine in particular has excellent T-cell immunity and has so by design. It's quite important to monitor these variants, as a particularly infectious and resistant one can take over very quickly, especially now that we're effectively applying selective pressure through natural infection and vaccination. So B1351, as you see from the yellow area in this graph, went from non-existent to becoming the dominant variant in South Africa in a matter of 8 weeks. Now it's nearly the only circulating variant in that country. And disturbingly, B1351 has been found circulating in 36 of 50 US states and territories and across Europe. Several variants containing also the key mutation of position 484 have also been reported in the United States, starting in New York City. Beyond this important high-level protection of the vaccine against variants, the J&J vaccine has other features that allow it to play an essential role in the effort to contain COVID-19, both in the United States and globally. Our vaccine is the only authorized vaccine that uses a single-dose regimen. The benefits of this dosing cannot be overstated, considering the urgent mass vaccination campaign we're in the midst of. Whether it's in rural communities that lack access to healthcare, or transient populations like farming communities that may not be able to in the same location for a second dose, the one-shot benefit is critical. The vaccine also has a relatively quick onset, with vaccine efficacy against severe disease seen as of day 7 in the pivotal trial. Available immunogenicity data from the phase 1 study up to date 115 are in line with the historical platform data, and thus very promising with regards to durability of protection, also in the elderly. And last but certainly not least, the vaccine is compatible with existing vaccine distribution channels and can be stored for 3 months at normal refrigerator temperatures. What this means for achieving vaccine goals should not be underestimated. We have the ability to reach locations in the United States that other vaccines may not. These features of the vaccine allow vaccination of people that might otherwise not be able to or choose not to get a shot. That is, there may remain part of an unvaccinated segment of the population. All in all, the J&J vaccine plays a critical and complementary role to other vaccines. To be explicit, one concern is that absent or a restricted J&J vaccine does not just delay the full vaccination of the US population, but potentially leaves unvaccinated a proportion of the US population. And if the unvaccinated population is large enough, we put at risk reaching herd immunity in the US. And the global context is also critical here. As global mobility resumes, there's no question that US residents remain vulnerable so long as large swaths of the globe remain unvaccinated and variants therefore continue to emerge. And for many parts of the globe, the single dose and easy transport and storage features of the vaccine are absolutely critical. A restriction in use here could have a negative impact on the success of achieving global herd immunity. The pandemic in the US will not truly end until the world has indeed achieved herd immunity. So we believe this perspective is critical for today's discussion. Let me now hand over to Joanne Waldstreicher.
Thank you for the opportunity to meet today. Safety is our highest priority, and we've been laser focused on identifying and understanding the very rare occurrence of CVFT and other thromboses together with thrombocytopenia, as well as working with FDA and other regulators on appropriate labeling to ensure awareness and guidance on diagnosis and treatment. There's been extensive outreach and communication of this event over the past week, which has stimulated a number of additional reports on this important and serious event. As we saw in Dr. Shimabukuro's presentation, there are 15 post-authorization cases of thrombosis with thrombocytopenia, all in women, most under 50 years old. Most of these cases were CVST, and some also had thromboses at other sites. I want to say that I use the word cases, but these cases are not just numbers to any of us, and we take them very seriously. These are people, and we need to understand and characterize their risks. In exploring the context, we will show the rates in relation to other background rates. This is meant to be a general comparison of orders of magnitude of risk, not a direct comparison. So let me now place this into context. When we look at the cases reported post-authorization, there have been 15 cases reported among nearly 8 million people vaccinated. That gives a reporting proportion of 1.9 cases per million people. To compare this to the background rate, we did an initial focus of CVST with thrombocytopenia. Our analysis of 5 large observational U.S. healthcare databases that included over 60 million people estimated the background rate of thrombosis CVST with thrombocytopenia to be approximately 0.1 cases per million. The evaluation of other thromboses with thrombocytopenia is ongoing, but many of those cases are confounded by other diagnoses such as cancer and other illnesses. In reviewing the interim Brighton criteria and speaking with experts, as well as evaluating the data thus far that we see in healthy people, we recognize that this is likely extremely rare. Here we show the observed cases of thrombosis with thrombocytopenia per million people on the bottom in comparison to other vaccine-related adverse events on the top of the line. By no means do we mean to equate all these events with the seriousness of thrombosis with thrombocytopenia, but we felt it was important to note the frequency of these events, such as intussusception for rotavirus vaccine, immune thrombocytopenia associated with MMR, Guillain-Barré for H1N1 vaccine, and anaphylaxis in the pre-COVID era from vaccines, all of which range from approximately 1 to 70 cases per million people. The fact sheet agreed upon with FDA describes that the clinical course of thrombosis with thrombocytopenia after our vaccine shares features with autoimmune heparin-induced thrombocytopenia. This is estimated to occur between 1,000 and 50,000 cases per million heparin users, which is a different patient population than the vaccinated population. Heparin-induced thrombocytopenia, as you heard earlier today, can be associated with life-threatening thrombosis, and it also requires proper diagnosis and, importantly, consideration of alternatives to heparin, as with thrombosis and with thrombocytopenia. Let me now turn our focus to women under age 50. Amongst the 1.9 million women in this group, there were 13 cases, which is a reporting proportion of 7 cases per million women. To contextualize the risk for women under age 50 further, we look to consideration of the labeling and known labeled association of oral contraceptives and thromboembolic events, including stroke, myocardial infarction, and CVST. Again, I want to emphasize that by no means are we equating these types of events with one another. However, I use this as an example of where there are options for healthy younger women, each product with its own benefit risk and product profile, among a variety of other options, each with unique characteristics, such as long-acting injections, transdermals, different hormonal generations, and even non-hormonal methods, and each with risks that are addressed with warnings in product labeling and clear information in the patient information leaflet. Ultimately, the risk of thrombosis with thrombocytopenia should be put into context with the benefits of vaccination. This slide is a benefit risk assessment in adults 18 years of age and older. Based on the efficacy data from the randomized pivotal trial and the reporting rate from spontaneous reporting, we could expect that if 1 million people in the United States were vaccinated with the J&J single-shot vaccine, there would be over 2,000 fewer deaths and 6,000 fewer COVID-related hospitalizations on the benefit side, and at the same time, approximately 2 additional cases of thrombosis with thrombocytopenia. When we look at the benefit risk assessment in women under 50 years of age, if 1 million women in the U.S. were vaccinated, there would be 2,600 fewer COVID-related hospitalizations and 116 fewer deaths on the benefit side, and approximately 7 more thromboses with thrombocytopenia cases. Janssen fully supports the public health recommendations from regulatory agencies and medical societies to create general public awareness, as well as to provide specific guidance for medical professionals regarding this very rare but serious event following vaccination. We're committed to active follow-up to identify and investigate all cases and have an ongoing effort to fully understand the pathophysiology. Our observational study protocols are designed, and we are examining the feasibility of multiple databases, not only from the U.S., but also from the rest of the world. We fully support the guidance from the CDC and professional societies that outline awareness, education, diagnosis, and treatment. In addition, Janssen is implementing several initiatives, including healthcare professional and consumer support, professional education, posting fact sheets on our websites, and a 24-hour global hotline. We absolutely agree with the FDA on the implementation of a warning within our label and patient and physician fact sheets describing this very rare event, including how it can be identified early and diagnosed and treated. To that end, here is the language from our label that we have agreed upon with the FDA regarding thrombosis with thrombocytopenia. In addition to the description of the event, healthcare professionals are alerted in the fact sheet to the signs and symptoms of thrombosis with thrombocytopenia in individuals who receive the COVID vaccine from Janssen. It also states that symptoms began approximately 1 to 2 weeks following vaccination, and that most people were females aged 18 through 49, and that some cases have been fatal. Healthcare professionals are also directed to the published American Society of Hematology considerations relevant to the diagnosis and treatment. In addition, the patient fact sheet contains a clear warning and instructions. J&J strongly supports this enhanced labeling. In conclusion, the benefits of Janssen's effective, single-dose, and easy-to-use COVID vaccine continue to outweigh its risks. We continue to face a deadly pandemic, and existing and emerging variants are making containment even more challenging globally. Every day matters in our campaign to control the virus, and we need all options available as the effects of COVID infection can be both serious and long-term. The Janssen vaccine offers many benefits in this context, including early efficacy and including demonstrated efficacy in South Africa with the B1351 variant predominating, and simplicity in terms of its single-dose administration, shipping, storage, and handling. As such, the vaccine provides access to people who are underserved, as well as certain populations that typically face barriers to healthcare services. As Dr. Mammon mentioned, our concern is that with the restriction, it's not just about delaying vaccination, but potentially leaving unvaccinated a portion of the United States. And if the unvaccinated population is large enough, we put at risk reaching herd immunity. It's also important to recognize that decisions in the U.S. have an impact globally, and with delayed vaccination and potential increased vaccine hesitancy, variants that emerge globally ultimately impact the U.S., especially as global mobility resumes. A restriction in use here could have a negative impact on the success of achieving global herd immunity, both here in the U.S. and globally. The pandemic will not be over until a large segment of the global population is vaccinated. We recognize that there is a very rare but important and serious risk, and J&J fully supports increased awareness, education, and enhanced labeling. With these efforts, we aim for earlier recognition and making this event more identifiable, diagnosable, treatable, and with improved chances for successful outcomes. In summary, we believe the J&J COVID vaccine is central to the effort to end the pandemic. Thank you, and we look forward to answering your questions now, as well as any questions you may have during the discussion.
Thank you very much for those presentations. We'll now open this up to questions with the voting members' priority initially. Not seeing any hands. Liaisons, do you have any questions? Again, I'm not seeing any hands. All right. The speakers will be on the call, and if there are any questions, Dr. Shaw, I'm sorry. Go ahead, please.
Thank you, Dr. Romero. Just a brief question for our colleagues. Given their experience with other adenovirus vectors, other adenovirus vaccine vectors, have we seen cases of similar thrombosis with thrombocytopenia?
This is Mathai Mammon. I'd like to ask my colleague, Johan van Hoek, to comment on your question, Dr. Shaw.
I think it's good the vaccines of your day. With regard to your question, why indeed there are similarities between the other vaccine, which is reported in Europe, the AstraZeneca vaccine, and ours. It is important also to note that there are quite some important differences between both vaccines. That relates both to the vector that is used. The one is a chimp adenovector. The other is a human adenovector, and we do know that they belong to different species, and also it has been demonstrated that there are differences in host cell receptor and also interactions with other host factors, which could lead to potentially differences in innate and adaptive immune responses. There's also differences in the transgene that has been used. We do have a transgene that is stabilized with a knockout of the S protein, a fusion in the region, a deletion in the fusion region, and also we have it stabilized with 2 mutations, which is not the case for the other transgenes. So at this stage, while indeed there are similarities, and certainly the clinical picture is very similar, at present there seem to be some differences also in the reported frequency, although we realize it's still early days. And so we think it is somewhat premature today to conclude that we can automatically extrapolate all the findings from AstraZeneca to the Janssen vaccine. But we do recognize that the clinical picture is very similar. So there might be a difference in frequency or eventually mechanism.
And sir, I apologize for the lack of specificity in my question. That's my fault. I was thinking of other COVID vaccines that utilize an adenovirus vector, such as the Sputnik vaccine, and then other non-COVID vaccines that may also utilize an adenovirus vector.
We do have, we cannot comment on the Sputnik vaccine. We don't know exactly what has been reported for that one. With regard to other adeno-based vaccines, there are not that many licensed for the moment. We do have one vaccine, an Ebola vaccine, that also uses the same adenovector. We have looked in detail to the database that we have for that vaccine. Of course, we don't have the millions that have been vaccinated. We today have more than 200,000 people vaccinated with the Ebola vaccine, and we have not observed any of these phenomenon in that particular clinical trial setting. We also have other vectors in other vaccines in clinical development using the same vector, specifically an HIV vaccine, for which we have several thousand women also vaccinated today. Of course, the data and the numbers are limited, but also important to note that we have not seen any sign of a similar phenomenon in those trials.
Thank you. Dr. Alt.
I was actually going to ask the very same question that Dr. Shaw asked, so I'll put my hand down.
Thank you all. All right, we'll ask Drs. Mammon and Waldenstreicher to still be on the call, and if there are any other questions for them, we'll ask them to come forward. Thank you again for that presentation and answering those questions. So now we'll move on to the next presentation, the VAST assessment by Dr. Grace Lee, who is the ACIP VAST's co-chair and vice chair of the ACIP. Dr. Lee, please.
Thank you very much, Dr. Romero. You can move to the next slide. Thank you. I wanted to take a moment to place the events of the last week in context, particularly given the multiple work groups involved. The ACIP Vaccine Safety Technical Subgroup, or VAST, reviews vaccine safety data on a weekly basis. Risk assessment is a dynamic process, and we are constantly reviewing new data from all available vaccine safety surveillance systems in the US and have been since the first vaccine was administered. The ACIP COVID vaccines work group focuses on benefit risk assessment, placing the information about benefits and risks in the context of the ongoing pandemic in the US. And benefit risk assessment is also a dynamic process that is continually updated as new information emerges. Finally, the ACIP committee today deliberates on all available information in order to make recommendations for use. As we're doing today, we will revisit recommendations as new information emerges. Next slide. The VAST work group specifically reviews, evaluates, and interprets post-authorization COVID-19 vaccine safety data, serves as a central hub for technical expertise from federal agencies participating in vaccine safety surveillance, advises on analyses, interpretation, and data presentation, and provides updates to the ACIP COVID vaccines work group and the ACIP on COVID-19 vaccine safety. But more importantly, the reason we have such a focus on vaccine safety is to ensure that the public has confidence in vaccines and in the US vaccination program. Next slide. To date, VAST has had 17 independent meetings to review and discuss post-authorization vaccine safety data. In addition, VAST members have joined additional meetings of the COVID-19 vaccines work group for specific review and updates of safety data presented on those calls. Next slide. So I'll focus in on VAST's role in risk assessment and the thought process of the VAST work group. On April 12th, 6 cases of CVST with thrombocytopenia were identified as a rare but serious potential adverse event following the Janssen vaccine. We noted that risk factors for CVST with thrombocytopenia were not well understood, and the work group felt strongly that timely and transparent communication with healthcare providers and the public would be crucial to maintain confidence in the vaccination program. Next slide. Within 24 hours, CDC and FDA colleagues released a communication along with a safety pause to ensure that there was immediate awareness by clinicians, public health colleagues, and the public of this potential adverse event. Next slide. On April 14th, ACIP convened an open meeting to discuss CVST with thrombocytopenia to ensure transparency and public awareness. At that time, we reviewed reported cases of CVST with thrombocytopenia after COVID-19 vaccines, and we discussed the need for additional information to support evidence-based decision-making, including age and gender-specific risk estimates and an evaluation of the benefit-risk balance of using the Janssen vaccine in specific subgroups. Next slide. At the VAST meeting on April 19th, we reviewed the new Brighton collaboration definition of thrombosis with thrombocytopenia syndrome, or TTS, which you heard about today from Dr. Shimabukuro. That is a broader definition in use now. But having the standardized definition is critical to facilitate investigations across surveillance systems in the US and globally. So I just want to take a moment to recognize and thank the Brighton collaboration team for their quick work on this definition. VAST noted that the Health Alert Network, the HAN, was extremely useful for supporting enhanced case finding in the US. And at that meeting, VAST requested additional information on the age and gender-specific rates of TTS following the Janssen vaccine for further risk assessment. Data from the VSG and the VA rapid cycle analyses were reviewed, and no safety signals were identified for CVST, other thromboembolic disease, or thrombocytopenia following the Janssen vaccine, with over 200,000 doses administered at that time across the two systems. We also received an update from the WHO Global Advisory Committee on Vaccine Safety on TTS cases following the AstraZeneca vaccine. This included a better contextual understanding of the country-specific approaches to use of the AstraZeneca vaccine that are driven by those country-specific benefit risk assessments. Next slide. So at yesterday's joint VAST and COVID vaccines work group meeting, additional safety data were presented, and we discussed the risk for TTS following the Janssen vaccine was 7 per million doses in females less than 50 years of age, with the highest rate noted in women 30 to 39 years of age. In contrast, the risk for TTS was less than 1 per million in females 50 and older and males of any age. Other potential risk factors included obesity, oral contraceptive use, hypothyroidism, and hypertension, but we really don't have enough information to make a determination about whether they are true risk factors or associations. There were 3 deaths, 7 remained hospitalized, and 5 have been discharged home. And the group also discussed benefit risk assessment and the evidence to recommendations framework during that meeting. Next slide. So in summary, where we are today, we recognize that TTS is a rare potential complication potentially associated with the Janssen vaccine, and that the risks should be balanced against the benefits of preventing death, hospitalization, and thromboembolic disease associated with COVID-19 infection. VAST notes that the risk of TTS appears highest in females less than 50 years of age, and other risk factors for TTS are not yet well established. Risk mitigation strategies may include the following. Number 1, minimizing exposure in high-risk populations. Number 2, increasing awareness and ensuring timely diagnosis and management of TTS. And number 3, educating patients and the public about the benefits and risks of available vaccines. VAST will continue to monitor TTS, thromboembolic disease, and thrombocytopenia in all available vaccine safety surveillance systems. And we will continue to update the ACIP COVID-19 vaccines work group, the ACIP Secretariat, and the full ACIP committee on a regular basis. Next slide. I just want to take a moment to thank our VAST members for their continued public service and dedication to ensuring vaccine confidence in the U.S. And I also wanted to thank our intrepid CDC co-leads, Lori Markowitz and Melinda Wharton, and our ex officio and liaison representatives and administrative support team. I also wanted to personally thank all of our vaccine safety colleagues in the Immunization Safety Office and the vaccine safety investigators in VAERS, VSD, CISA, VSAFE, VA, FDA, CMS, and DOD for their round-the-clock work on vaccine safety since that first dose was administered in the U.S. on December 14th. Thank you.
Thank you. We'll now go on to presentation of thrombosis and thrombocytopenic syndrome, TTS, after COVID vaccines. Applying the evidence to recommendation framework. Presenting will be Dr. Sarah Oliver, who is the CDC lead for the COVID-19 work group. And before she begins, I want to thank her and her group for all the work they've done on this issue and on all the other issues that have come before us beforehand. So thank you very much, and please go forward.
Thank you, Dr. Romero. It has absolutely been a group effort over this last bit. So good afternoon. Next slide. Next slide. So for previous votes for the use of COVID-19 vaccines, we've used the evidence to recommendation framework. It's a structure used to to describe information considered in moving from the evidence to the ACIP vaccine recommendations. It provides transparency around the impact of additional factors on deliberations when considering a recommendation. Next slide. As a reminder, this is the policy question that was originally voted on for the Janssen vaccine. Should vaccination with the Janssen COVID-19 vaccine, 1 dose, be recommended for persons 18 years of age and older under an EUA? Next slide. This is the traditional evidence to recommendation framework used for previous recommendations. While it's helpful, it doesn't necessarily fit the type of questions facing ACIP today. Next slide. So we've adapted the evidence to recommendation framework to a risk-benefit assessment for the Janssen vaccine recommendations. We'll be using many of the same domains to lay out the information that we have available. First, for the public health problem, we'll be describing recent COVID epi, as this is what is most relevant to policy decisions made today. Then we'll provide a high-level review of other information that may inform the discussion, including thrombosis seen after COVID, an overview of CVST, the epidemiology of HIT, or heparin-induced thrombocytopenia, and what is currently known from the AstraZeneca vaccine. Then under benefits and harms, we'll discuss the benefits of the Janssen vaccine, harms associated with the Janssen vaccine, and our data on the benefit-risk assessment of COVID vaccines. Next slide. Then we'll discuss values and acceptability, including the intent to receive a 1-dose vaccine and how that has evolved over time. For feasibility, we have rapid recent surveys of jurisdictions over their use of the Janssen vaccine and the possible impact of Janssen vaccine policy options. And finally, we will highlight the possible impact the Janssen policy options may have on disproportionately affected populations. There are no data for resource use available to inform these discussions. And before we go in detail, now that we've highlighted what we're about to lay out, I just personally want to take a moment to thank the team of people who have done about 6 months worth of work in this last week to pull it together. Our data team, which conducted the risk-benefit assessment, and the modeling team, which helped provide the models to inform this assessment. So next slide. So first, describing the public health problem. Next slide. Again, this is what we'll be reviewing. Next slide. So this slide shows the trend in number of COVID-19 cases in the US. Through April 17th, we've had around 31 million cases. We had the rapid decline in cases since the winter, but with a stable to slight increase in recent weeks. Next slide. This is the daily death curve with over 560,000 deaths throughout this pandemic. Next slide. So this is a map of the US where each state is shaded by the most recent 7-day case rate, where the darker colors represent a higher incidence. Next slide. This slide highlights the epi curve from 2 states, one with a high incidence currently and rising, the other with a lower stable incidence. This is more to highlight that in a country this large, the epi of COVID is not the same everywhere. The pandemic is dynamic, and states can experience highs and lows at different times. Next slide. We also can't talk about recent epi without acknowledging the SARS-CoV-2 variants in the US. This slide shows circulating proportions and overall, and then the proportion of cases from variants of concern or variants of interest has increased. And you can see here in recent weeks, nearly half of the cases are the B117 variant. Next slide. We also have to acknowledge what is happening in the US with COVID is only a portion of the picture as well. This map shows a recent global incidence rates with a darker blue representing a higher rate in the last 7 days. Next slide. So again, the recent COVID-19 epidemiology will be the most informative for policy decisions made now and moving forward. So we're going to focus in on the epi since March 1st. Next slide. This slide shows the incidence by age and by sex. The highest incidence for new cases recently is actually females 18 to 29 years of age. Among adults less than 50 years of age, females have a slightly higher incidence compared to males, and this is reversed in the older populations. Next slide. This slide shows recent hospitalizations by age and by sex. Older adults have much higher rates of hospitalizations, with older males as the highest rate. Next slide. And finally, this slide shows mortality rates by age and sex, where older males have the highest rates and all younger adults have a lower mortality rate. Next slide. This slide shows the recent incidence by race and ethnicity. Disparities in incidence by race and ethnicity are noted. Next slide. This slide shows mortality by race and ethnicity. And again, you can see disparities by race and ethnicity noted. Next slide. So to summarize our COVID epi data since March 1st, for incidence, the cumulative rate is just over 700 per 100,000 population, where younger females have the highest incidence of new infections. For hospitalization, the cumulative rate since March 1st is 20 per 100,000. Most hospitalizations still occur in persons 65 and older, but it's worth noting that the proportion of hospitalizations occurring in this population is declining. For mortality, the cumulative rate since March 1st is 3 per 100,000. Again, where most COVID deaths are occurring in older populations, but the proportion occurring in this population is declining as well. Next slide. So next, we'll briefly cover the epi of several other relevant conditions. We've heard throughout today about CVST, as well as splanchnic vein thrombosis, or SVT. The overall incidence of CVST is around 14 to 28 per million. There is a wide range, and many of the differences in incidence that you may have heard from last week and across presentations this week is accounted for by this next fact, that the incidence has been steadily increasing in recent years. It's thought that this could be related to better detection by wider use of more advanced neuroimaging. CVST rates are higher in young women, and in traditional CVST, risk factors are identified in most cases. The mortality rate is 5 to 10%. Splanchnic vein thrombosis incidence is higher, with around 80 to 180 per million, with a higher incidence among men. And it's worth noting that the incidence with thrombocytopenia of either of these conditions is much lower than the incidence without thrombocytopenia. Recent estimates for CVST with thrombocytopenia are around 0.7 to 1.6 per million. But again, estimates for this condition are impacted by time and overall increases in CVST. Next slide. So heparin-induced thrombocytopenia, or HIT, occurs in 0.5 to 1% of patients exposed to heparin. This translates to somewhere around 20 to 45 per million in the US population. Of patients with HIT, thrombosis occurs in 20 to 64%. As we've heard described earlier today, it's an immune-mediated event with antibodies directed against platelet factor 4. There are risk factors for transitioning from HIT to HIT with thrombosis, which includes genetic factors, a lower platelet count, higher titers of PF4 antibodies, and prior surgery. Next slide. So CVST can occur after COVID-19, although it is very rare and in less than 0.1% of hospitalized COVID patients. Translating this to overall numbers, it could be estimated to be around 5 to 6 cases of CVST per million SARS-CoV-2 infections. CVST plus thrombocytopenia in COVID patients appears to be extremely rare. Any clots with COVID appear to be pathologically different than TTS after COVID vaccines. There was a preprint study that looked at PF4 antibodies in a history of patients with confirmed COVID. Overall, 90% of those were negative for PF4 antibodies, and all were negative on functional assays. This included patients who had thromboembolic events with their COVID infection. Next slide. There was one study described or mentioned earlier today that looked at electronic health records to look at CVST and portal vein thrombosis in COVID patients. They showed a low incidence of CVST in any 2-week period, but 2 weeks after COVID estimated an incidence at 39 per million hospitalized COVID patients. Cases of CVST were rarely seen after receipt of mRNA vaccines in this administrative data. However, there are limitations to interpretations of this administrative data. They were unable to assess the incidence of CVST after either advector vaccine, either the Janssen or the AstraZeneca vaccine, and did not provide rates of CVST plus thrombocytopenia. So it limits the direct ability to compare rates of CVST plus thrombocytopenia seen after COVID vaccines in the US or Europe. Next slide. So then briefly walking through what we know about thrombosis with thrombocytopenia after the AstraZeneca vaccine in Europe. Around 160 cases of CVST, 53 cases of splanchnic vein thrombosis were reported through early April in Europe. The EU estimates this to be around 10 cases per million, but acknowledges a higher incidence in younger adults. Their cases, again, were mostly in women less than 60 and within 2 weeks of receiving the first AstraZeneca vaccine dose. The EMA concluded that the benefit risk was still favorable to use the vaccine, and a causal association was likely. No specific risk factors to date, but they acknowledge that the epi may be related to vaccine delivery across Europe. They also added a warning for unusual blood clots and low platelets to the label as a side effect. Next slide. The UK recently provided updated numbers through mid-April with 168 reports of blood clots with low platelets, 72 cases of CVST. The male-female split seen is slightly more even with a broader age range, was discussed in our first presentation today. They note a rate of 7.9 per million doses. The UK has also decided that the benefits continue to outweigh the risk, but JCVI, or the UK's version of ACIP, recommended that persons between 18 to 29 years of age at low risk of infection be offered other vaccines. Next slide. Earlier this week, the EMA also met to review 8 of the cases of TTS after the Janssen vaccine reported in the US. They felt that the cases were very similar to cases occurring after the AstraZeneca vaccine. Similar to AstraZeneca, they concluded that a warning should be added to the product label. However, they also emphasized that it was a rare event, the benefits outweighed the risk, and emphasized the importance of healthcare provider awareness. Next slide. So this slide just pulls together what we've presented over the past several slides into a single slide, with the COVID outcomes in blue on the left, HIT, CVST, and CVST with thrombocytopenia rates across the top, and then the rates of TTS after AstraZeneca vaccine are shown in the box. Next slide. So now moving to benefits and harms. Next slide. So again, we'll highlight the benefits of the vaccine, harms, and our overall risk-benefit summary. Next slide. The phase 3 trial of the Janssen vaccine demonstrated efficacy against symptomatic, lab-confirmed cases of COVID with an overall estimate of 66.3%. Efficacy against hospitalization was 93%, and higher efficacy against severe outcomes was noted. Efficacy against severe disease remained high in various world regions, suggesting protection against severe disease with variant strains. Next slide. This was a slide shown from our former ETR presentation with a plot showing similar efficacy across age, sex, race and ethnicity categories, and among those with and without underlying medical conditions, at least 14 days after vaccination. Next slide. There are also benefits beyond efficacy. This vaccine is able to be shipped and stored at refrigerator temperatures and is a single-dose series, which may make it easier to reach some disproportionately affected groups. Next slide. Regarding possible harms of the Janssen vaccine, Dr. Shimabukuro described this in detail, so I won't go through it all again, but highlighting the findings here. Nearly 8 million vaccines administered, with 15 confirmed cases of TTS, with a rate of 7 per million in women 18 to 49 years of age, almost 1 per million in women 50 years of age and older, and none in men noted in post-authorization surveillance, although we do acknowledge that one case occurred in a man in the clinical trial. Next slide. So now I'm going to walk through our formal risk-benefit analysis. We're going to show this from two different perspectives. As ACIP thinks through these policy decisions, you'll need to think through the population-level risk-benefit and an individual-level risk-benefit. Therefore, we'll be presenting results from two different analyses that look at each of these perspectives. First, for the population level, we'll quantify COVID infections, hospitalizations, and deaths averted under different assumptions about resuming Janssen COVID vaccines over a 6-month period. Then we'll use that data to quantify population-level, age-specific benefits and harms of resuming vaccination with Janssen COVID vaccine. Then for the individual-level risk-benefit analysis, this was a more direct patient-level evaluation looking at a 1-month period to quantify the direct age and sex-specific benefits and harms per million Janssen vaccine doses. Next slide. So first, for the population level, objective of the modeling was to quantify COVID-19 infections, hospitalizations, and deaths under different assumptions about resumption of the Janssen vaccination. In all scenarios, we assume continued use of mRNA vaccines. There were 5 scenarios evaluated: that the Janssen vaccine was not resumed, that it was resumed starting tomorrow for all adults 18 years of age and older. This was then at 50% of a pre-pause administration rate or 100% of a pre-pause administration rate. Then that the Janssen vaccine resumed tomorrow in adults 50 years of age and over only, again at a 50% pre-pause administration rate or 100% pre-pause administration rate. Next slide. Our modeling team developed a compartmental model used to simulate incident infections, hospitalizations, and deaths over the course of the epidemic in the US. This is an updated version of the model, which has previously been shared with ACIP and is stratified by age, essential worker status, and underlying conditions. The model was recalibrated with incidence data through spring of 2021 to be consistent with the ensemble from round 4 of the multiple model scenario hub. We modeled age group specific vaccination coverage through April 2021 that also accounted for age group specific vaccine hesitancy when calibrating anticipated coverage. We modeled 2 types of vaccines, mRNA and Janssen vaccines, with VE estimates informed by the clinical trials and assumed to develop 14 days after administration. We assume no loss to follow-up with mRNA second doses, no delays in administration, and we also assume no waning immunity over the future 6-month time horizon. Next slide. So this slide looks at time to complete vaccination for all intending adults. During and after the vaccine pause, administration of mRNA vaccines continues at the same rate as the week before the pause began until demand is fully met. If use of the J&J vaccine, the Janssen vaccine, is not resumed for any ages, then it takes approximately 14 days longer to meet all vaccine demand. However, in practice, the pace of administration may slow as those experiencing barriers to access and/or greater hesitancy may comprise a greater share of those unvaccinated. Next slide. So this graph illustrates how the results of varying the resumption strategy might be influenced by the population vaccinated. Hold on, I may have skipped. Yes, sorry. This model was calibrated to be consistent with the ensemble from round 4 of the multiple model scenario hub. We modeled both low transmission scenarios, shown in blue, with a higher and longer sustained compliance with non-pharmaceutical interventions, and a moderate transmission scenario, shown in black, with non-pharmaceutical interventions easing more quickly over time. Next slide. So then this graph illustrates the results of varying the resumption strategy may be influenced by the population vaccinated and administration rate under this moderate transmission scenario. So the blue bars show the percent reductions in infections, hospitalizations, and deaths, assuming resuming vaccination in persons 18 years of age and older. And the green bars show a much lower percent reduction in those outcomes when limiting Janssen vaccine to person 50 years of age and older. Additionally, the lighter and darker bars show the difference in model results depending on administration rate, with 100% administration rate resulting in greater reduction than the 50% administration rate. Next slide. So now this graph on the right shows the model results under a low transmission scenario with a slightly lower percent reduction. And the outputs from the model on this slide were used in the population risk benefit calculations that follow. Next slide. So again, we use the outputs of that model just shown to quantify the age-specific risks and benefits of resuming vaccination with Janssen COVID-19 vaccines, including the risks of TTS and the benefits, including prevention of COVID-19 related hospitalizations, deaths, and ICU admissions. Next slide. From the model, we use the numbers of hospitalizations and deaths due to COVID-19, the number of persons estimated to receive the Janssen vaccine after the pause based on a variety of conditions, including, again, low and moderate transmission, 50 and 100% of the vaccination pause, and then the resuming vaccination in over 18 and over 50%, in over 50 years compared with no resumption. The estimates are modeled for a 6-month time horizon. And additionally, data from the CDC pandemic planning scenarios on age-specific proportions of hospitalized cases admitted to the ICU, CDC counts of the observed TTS by age, and numbers already vaccinated with the Janssen COVID-19 vaccine data before the pause were used. Next slide. So here we show the scenario where vaccination is resumed in persons 18 and older using the conditions of moderate transmission and vaccination with the Janssen vaccine at 50% administration rate before the pause. These are the number of additional benefits for the entire population and harms compared to not resuming vaccination with the Janssen COVID-19 vaccine. In the scenario of resuming vaccination for all persons 18 years of age and older, we expect 2 TTS cases among 50 to 64-year-olds and 24 cases among 18 to 49 years, and a many greater times reduction in death and ICU admissions. Overall, if vaccination with Janssen COVID-19 vaccine were resumed in all adults, the model predicts that 26 cases of TTS will occur among the 9.8 million persons vaccinated, and that about 2,200 deaths and 1,400 ICU admissions would be prevented. Next slide. So now on the right, the scenario of resuming vaccination only for persons aged 50 and over. There are 2 cases of TTS predicted, again, only among 50 to 64-year-olds. The number of deaths and ICU admissions prevented are much smaller than the scenario of resuming vaccination for all adults, but the benefits still outweigh the smaller risks. The model predicts 2 TTS cases in 3.6 million vaccinations, which would prevent around 250 deaths and 800 ICU admissions. Next slide. So summarizing the risks and the benefits for all the scenarios included in the model, considered suggesting upper and lower bounds of population effects for resuming Janssen COVID-19 vaccines. When resuming vaccination among all persons at least 18 years, we expect 26 to 45 TTS cases, depending on vaccine uptake, and expect between 800 and 2,500 fewer admissions and between 600 and 1,400 fewer deaths compared to not resuming vaccination with the Janssen vaccine. When resuming vaccination among only persons 50 years of age and older, we anticipate 2 to 3 TTS cases and between 300 and 1,000 fewer ICU admissions, and between 40 and 250 fewer deaths. With this approach to assessing the risks and the benefits, we note that the benefits of vaccination apply to the whole population over a 6-month period and result from both direct and indirect effects, whereas the risks only pertain to the vaccinated persons. Next slide. So now we're going to move to the second level that we talked about considering. We just showed the population level benefits and risks, and next we're going to move to the individual level benefits and risks. The population model did not inform the direct individual benefits and harms for vaccination of that individual and was not able to look at sex-specific effects. So we used a simpler direct estimation approach to explore risks and benefits for females and males in different age groups per million Janssen vaccine doses. The calculations were based on recent age-specific incidents of hospitalization for COVID, VE against hospitalization observed in the trials, total Janssen vaccines to date, the number of persons already vaccinated, and are calculated assuming a 30-day period of not being vaccinated. Next slide. For females aged 18 to 49, we estimate that for every million doses of Janssen COVID-19 vaccine administered, we would expect 13 cases of TTS and would expect to prevent 12 deaths, 127 ICU admissions, and 657 hospitalizations due to COVID in 1 month. Next slide. On the right, we see the estimation for persons for females 50 years of age and older. There are only 2 cases of TTS per million vaccination doses, but we would expect to prevent nearly 600 deaths and over 1,000 ICU admissions and nearly 5,000 hospitalizations due to COVID. Next slide. In males aged 18 to 49, we incorporated the 1 TTS case that was observed in the phase 3 trial in this estimate. Per 1 million vaccinations, we might expect a small number of TTS cases and would expect to prevent 11 deaths, over 100 ICU admissions, and 600 hospitalizations due to COVID. Next slide. So then in males 50 years of age and older, we expect 0 cases of TTS and could avert around 700 deaths, 1,500 ICU admissions, and 5,000 hospitalizations due to COVID. Next slide. So the population and individual approaches to the risk-benefit analysis offer complementary information on the risks and benefits of resuming vaccination and in which age groups. The population approach takes into account both direct and indirect or herd effects of vaccination. It incorporates the availability of different vaccines, simulates incidents, hospitalizations, and deaths over the course of the pandemic, and makes estimates on all outcomes over a 6-month time horizon. This approach shows a large population benefit of vaccination relative to a rare occurrence of TTS. Next, the direct individual level approach estimates the benefits and risk to vaccinated individuals and only considers getting a Janssen vaccine versus not getting any vaccine over a short 1-month time horizon. The analysis shows a positive balance of benefits and risk for all ages and sex groups. However, the stratified results suggest that the relative balance and risks for individuals varies by age and sex. Next slide. So now moving to values and acceptability. Next slide. We'll present intent to receive a 1-dose COVID vaccine and intent to receive a Janssen vaccine over time and the effect on overall confidence. Next slide. So prior to authorization of a Janssen vaccine, we conducted a survey that asked if there was a 2-dose or a 1-dose COVID vaccine available, which would you choose? And this survey was conducted in February of 2021, prior to the pause. Overall results were presented back in March 1st when ACIP voted on the use of the Janssen vaccine. However, we now examined this data by age, sex, race and ethnicity, and income. Overall, most responders said that they would be willing to take either vaccine offered to them. However, 6% of individuals said that they exclusively preferred a one-dose vaccine. There were no differences in the proportion that preferred a one-dose vaccine by age, sex, or income, but there were significantly more Hispanic than white respondents that exclusively preferred a one-dose vaccine. Next slide. So there have been several other surveys conducted over the past 1 to 2 weeks. At the time of this last survey, only 37% of Janssen respondents called the Janssen Covid vaccine safe. That was announced, this was after the pause was announced. This represents a drop of about 15% over 2 to 3 days. Another survey found that Americans are less likely to prefer a Janssen vaccine, with 13% decline in preference for the vaccine, which ranged from 9 to 25% across age and race categories. Next slide. This slide shows a figure from one of the surveys demonstrating a willingness to get each vaccine over time. You can see in recent weeks, the willingness to receive a Janssen vaccine has declined substantially. Next slide. So then trying to look at overall vaccine confidence. One survey found that the drop in vaccine confidence did not appear to extend to the Pfizer or Moderna vaccines, where most felt that they were safe. A recent poll did not suggest reduction in the intent to be vaccinated, and instead found that people were 40% more likely to receive a vaccine compared to 1 month ago. One survey did find a proportion of those who remain unvaccinated may be less inclined to receive a COVID vaccine after the pause, regardless of the brand. Next slide. So moving to feasibility. Next slide. We were able to conduct a rapid survey of each U.S. jurisdiction for the use of the Janssen vaccine and the impact of the Janssen vaccine pause and potential recommendations. The figure on the left shows where jurisdictions had been using the Janssen vaccine. You can see that many jurisdictions were using this vaccine in specific populations, including persons experiencing homelessness, homebound populations, and incarcerated individuals. The figure on the right shows settings where jurisdictions were using the Janssen vaccine, which included mobile vaccination clinics, emergency departments, and student health centers. Next slide. We then asked jurisdictions for the impact if the Janssen vaccine were only recommended for specific populations. Summaries of responses are presented here. Jurisdictions felt that they may need to reconfigure some of their vaccination sites, update scheduling tools, and may have difficulty serving disproportionately affected populations. Many stated that providers may need to carry multiple vaccines. Health departments also expressed concerns about communicating changes to the public, and again highlighted the difficulty serving disproportionately affected populations, for populations who are at risk for not returning for a second dose, race and ethnic minorities through mobile vaccination, and in rural or hard-to-reach areas. Next slide. So this slide shows jurisdictional thoughts for an impact if the Janssen vaccine was no longer recommended. Jurisdictions were primarily concerned around second dose management if only 2-dose vaccines were available, and equity concerns. Janssen vaccines provided flexibility for jurisdictions to avoid second dose management, run mobile clinics, and fully vaccinate people who may need to complete a series quickly. Jurisdictions noted some individuals expressed a preference for a Janssen vaccine, and increasing challenges to reach homebound, transient, or rural populations. Next slide. So finally for equity, next slide. This is from the same jurisdictional survey, and most was highlighted before, but we're highlighting here, again raising concerns from the jurisdictions around equity. Jurisdictions continued to mention 4 populations at risk of disproportionate impact. persons experiencing homelessness, homebound populations, incarcerated individuals, and migrant or seasonal populations. It was felt that if the Janssen vaccine were no longer available, these populations may be disproportionately impacted. I won't go through and read the quotes, but they're here and highlight the concerns and disparities from various jurisdictions. Next slide. So now we'll talk about the policy options for the use of the Janssen COVID-19 vaccine. Next slide. This slide was showed at the ACIP meeting last week. There's a spectrum of options for the Janssen vaccine. First, ACIP could decide that the risks outweigh the benefits and vote to not recommend use of the Janssen vaccine due to these safety concerns. ACIP could decide that the benefits outweigh the risks overall and recommend use of the vaccine in all adults 18 years of age and older. There's also an option in the middle of the spectrum, recommending use of the Janssen vaccine in some populations. Next slide. So these were the 4 policy options discussed by the work group. First, recommend against use for all persons. Next, reaffirm recommendations for all ages and sex in the content of the FDA warning statement within the EUA that was mentioned earlier. Next, recommend vaccination for only adults 50 years of age and older. And finally, reaffirm recommendation for use, but women less than age 50 should be aware of the increased risk of TTS and may choose another COVID-19 vaccine, such as the mRNA vaccines. Next slide. I'll walk through some highlighted pros and cons for each of the 4 options that were discussed. First, to recommend against use in all persons. A pro would be that there would be no further development of cases of TTS after the Janssen vaccine. However, the cons would be that it would remove choice from individuals. It could lead to excess COVID-19 cases and deaths, and could disproportionately impact at-risk populations who have barriers to access or difficulty returning for a second dose. Next, the population to reaffirm for all ages and sex in the setting of the FDA warning. This would allow for flexibility and choice and allow for use of the vaccine in harder to reach populations. However, it would place the burden on the individual to understand the risk and on health departments or providers to convey this risk. It may lead to more cases of TTS. And it's worth noting that at-risk populations for COVID-19 are likely at risk for barriers to identification and prompt treatment for TTS if it should occur after the vaccine. Next slide. So next, to recommend vaccination for adults 50 years of age and older. This would remove a vaccine from the most at-risk population and thus sharply reduce expected TTS cases. In addition, a single age cutoff is easy to communicate. However, it would likely be difficult to implement, where vaccination sites could need to stock 2 different vaccines. It would remove an option in a population that's actually at lower risk, men less than 50 years of age, and could disproportionately impact at-risk populations. And finally, the 4th policy option, to reaffirm recommendations for use, but recommends that women less than 50 should be aware of the increased risk and may choose another COVID vaccine. This would allow for flexibility and choice and allow for use in the vaccine in hard-to-reach populations while still acknowledging the risk in young women. However, it could still be difficult to implement, again with sites possibly needing to stock 2 vaccines, and could be difficult to communicate. Next slide. So next I'll highlight quickly a summary of the workgroup discussion that occurred yesterday. There was concern by many that a detailed personal level discussion of the risk-benefit balance is difficult in many of our current vaccination settings, such as mass vaccination clinics. Recommendations that may require vaccination sites to require 2 types of vaccines is difficult to implement. Access to vaccines for hard-to-reach populations remains important, and that the risk-benefit balance may change as the pandemic evolves and the risk of COVID-19 changes. Next slide. So again, the work group discussed benefits and concerns with all policy options. There was no single policy option as a clear choice from the work group. However, many on the work group appreciated the flexibility with broader recommendations for use, but acknowledgement of the elevated risk in women less than 50 years of age as the fourth option that was previously shown. Next slide. As a reminder, this is the previous vote. The Janssen COVID-19 vaccine is recommended for persons 18 years of age and older in the US population under the FDA's emergency use authorization. So our question proposed to ACIP, given the review of the benefits and risks presented here, what recommendation does ACIP feel is appropriate for the use of the Janssen COVID-19 vaccine? Next slide. And I'll leave these up for discussion. Turn it back over to you, Dr. Romero.
As always, thank you for a wonderful, detailed, and lucid presentation. So this is now open for discussion. We'll begin with the voting members. Let me pull up my screen here. Are there any comments or questions? Dr. Bernstein.
Spectacular presentation. Thank you, Sarah. The question that I have is, oh, I'm sorry, I don't know I'm echoing.
We hear you well.
Okay, good. I was interested with the fourth option that says reaffirm recommendations for use. Women aged less than 50 years should be aware of the increased risk of TTS and may choose another COVID-19 vaccine, that is mRNA vaccines. I was interested to know some more detail on how that women aged less than 50 years would become aware of the increased risk of TTS. I asked at last week's meeting, adding a warning to the EUA that's already 6 or 8 pages long. I was concerned that such a warning would be overlooked or missed, and I was wondering what's being proposed to convey that important information.
Dr. Bernstein, this is Amanda. I can respond to that. Yes, we awaiting ACIP's recommendation. We will have prepared, if this is the direction ACIP goes in, we will have tools both for providers as well as a public education to inform women and providers about not only the risk, but what signs and symptoms to be aware of if you do choose to get COVID vaccine. And we'll do this through, high level infographics and other ways to convey this information in a much simpler way than will be communicated in the very comprehensive and complete patient FAQ form in the EUA.
Thank you.
Dr. Sanchez.
Thank you. Sarah, that was wonderful as always, and thank you. I understand the complexity a lot. I understand the having a permissive acceptance of the vaccine would be like a shared decision making that we have been doing for other vaccines. I actually was very interested and was going to ask even the Johnson & Johnson officer, Dr. Alstracher, if they had information on the acceptance of the vaccine after all of these cases have been reported in the media and in the literature. And I see that it's down to 19%, so thank you for that information. My concern is that that currently most, I think very few places, at least that I'm aware of, have options for different vaccines. And if we only have the Janssen vaccine available, there will be a lot of people who may, if that 19% is true, they will choose not to take that one. So I think that we're going to end up having to have other vaccines. And other vaccines are available, as we all know. I also understand the rationale for comparing this vaccine to an unvaccinated population and the risk benefits with that. But the fact is that we do have other vaccines that, at least at this time, don't have this. We have not seen the TTS. So I remain concerned. I understand the shared decision-making and having that option for individuals, but I also find that it would be very interesting to see how many, after we actually inform them of this potential risk, if that number, 19%, might even go down further. And so I think it's a really difficult decision, but I also think that that if it is approved and we recommend against, in the use of any of these, I think that we really will need to have choice available at different jurisdictions. Thank you.
Thank you, Dr. Sanchez. Dr. Long.
Yes, first, so that I understand the very fantastic modeling. First, on the population level, it was quite compelling. Is that all based on your assumption that it would take 14 days longer to immunize the population than if you had this vaccine available for all? I just missed that part of it. And similarly, as Dr. Sanchez asked, the decision to compare individual direct benefits of the vaccine versus none, is that because you wanted us to understand what would happen if an individual refused other vaccines?
So I'll take the second question first. I want to highlight the population level model included mRNA vaccines, our current and projected supply over the next 6 months. So the population level really kind of was the situation as it is in the US. The individual patient level model really tried to take into account that the thought that there might be a time period weighing the risks and the benefits if that individual is unable to get access to another vaccine in a short period of time. So I'll say that the population level looked over time with the mRNA vaccines, that individual level, that's the reason it was a short time horizon, because we assumed that the delay would be low. Then back to your first question, that 2-week, it was kind of a 2 to 4 week, that was actually an output from the first model, assuming kind of the current situation right now in the US. So the time delay was an output of the model, not an input to the model. And Rachel is on, our fantastic modeler. Did I say it correctly, and do you have anything else to add?
That's a great response, Sarah.
Thanks. This is Amanda.
Can I ask one more question along this line?
You have the floor.
Thank you. Why did you just not at all think about giving this just to males under who are younger? That was about 3 double negatives, but I bet you get it.
I think I understand. When we had to choose the scenarios for the modelers to put in, that was based on the initial discussions from the work group last week, that an age-based recommendation is something that jurisdictions felt, could potentially be communicated and discussed, but having a sex-based recommendation would be both incredibly difficult to communicate and really difficult to implement. So when kind of push came to shove, that is why we chose to not to go with the age-based as opposed to the gender-based.
Could you just enlighten me a little bit about how someone declaring whether they're male or female would be so much more difficult to implement than declaring their age?
Dr. Long, this is Amanda. I think that some of the challenges, for example, if a family came, it's not around somebody declaring their gender and as much as it is where different groups of people access vaccines and the different types of places that would have to potentially carry multiple types. I think that the work group recognized the same challenges with the age restriction as well as with the gender restriction. And I think that, there was also, over the course of the week, there was, there have been reports of male cases that are under, that some of which may have been ruled out or whatnot, but I think that the risk in males is still not completely, while appears to be very low, there is still likely some risk. I want to say that I've been getting some reports that some of the listeners have been having trouble hearing some of the ACIP members, so please just speak closely to your mic. Thanks, sorry.
Next, Ms. Bata.
Thank you, and thank you, Dr. Oliver, for this incredible presentation. One of the things that I've been hearing since the pause has started has come from several of our BIPOC communities, our Black, Indigenous, and people of color communities, in that they perceive that the vaccine has been targeted to, the Janssen vaccine has been targeted to them, and we're already concerned about the number, the efficacy number, but they're also concerned that this could be a subpar vaccine in terms of safety. And so I think that's a piece that needs to be thoughtfully considered. And it may become irrelevant, but I didn't see the race. I know that we had race breakdown last week, but do we have any additional knowledge about the impact on race, ethnicity?
This is Sarah. I am not aware, and again can defer to Rachel, of any kind of specific model outputs from risk and benefit that we were able to obtain from kind of the population level at a race and ethnicity level. What I will say is it felt like that many of those concerns came out in the jurisdictional surveys as well, which I think communicate many of the concerns that you raised. Rachel, I'll go to you though quickly. Do you know of any race and ethnicity output that you guys were able to get from the model?
No, the models look at persons within an age group, and they do not distinguish between different races and ethnicities, what their contact patterns would look like, nor their risk of severe outcomes. So unfortunately, that's not an output of the model shown here.
Okay, thank you. And then what is the feasibility of vaccinators being able to have two vaccine products? And I'm thinking in terms of the upcoming availability of vaccines. In the past, we really haven't had much of a choice of what vaccine we're going to get. And can we accommodate that flexibility so that if we would do something like a reaffirmation of the recommendations, but notification to women under 50, could we have an option for them based on supply?
Ms. Bata, I can start, and then Dr. Romero can also join, but the distribution at the national level would continue to be the same, and it would be local decisions about where vaccines went to different sites. Dr. Romero?
So if you'll permit me, I'll throw my Secretary of Health hat on for the state of Arkansas. So this would be an implementation issue that would be dealt with at a local, statewide level, and we would have to adjust to that, and understanding that that is the case, we would probably have to flex to do so. Again, this is speaking only for my state, and if the representative from ASTHO would like to come and say anything, that would be appreciated also. But I think we can honestly accommodate a recommendation of that nature. So, Dr. Shah, if you wish, if you feel free to make a comment.
Thank you, Dr. Romero. Again, this is Nirav Shah, speaking on behalf of ASTO. It is feasible to accommodate multiple vaccines as well as prescribe different recommendations for individuals. But as Dr. Oliver noted in her exceedingly fantastic presentation, it does introduce logistics challenges of two natures. One is that it requires an adjudication of some decision. And in a time when we're trying to vaccinate as many folks as possible in any given period of hours, it might slow things down. Even a couple of percentage points may have an impact. The second, though, and what I've heard from pharmacists across states is that carrying multiple vaccines does introduce the possibility, small though it may be, but real, about the possibility of errors of different sorts happening.
And then this is one again, I just have one additional comment, and this is just in the context that we are looking at a very rare risk and the high benefit and the need to make all vaccine available to all of us, and that we need that flexibility. And so I would tend to look for the decision with the fourth consideration.
Thank you for those comments. Dr. Paling.
Yes, thank you. First, Dr. Oliver, thank you. This presentation, and I want to thank you and all of your colleagues for this great presentation and the huge volume of work that you have presented. I particularly am focused on recognizing that we are in the midst of an ongoing pandemic, and that in the population level, the benefits are far greater than the cases of TTS. And I also very much appreciate the many hard-to-risk populations, as well as the very important point that Ms. Bata shared about making sure we communicate and don't make people feel like the vaccine is targeted to any one group. The second point is I wanted to say thank you for the individual assessment, because that provides a very important component as I'm thinking about it. And when you look at the individual, for all groups you presented, the benefits clearly outweigh the risk, though there are differences in age groups, and particularly for women less than 50 years of age. And for all of those reasons, I am leaning towards the fourth choice as well. Thank you.
Is someone speaking? We don't hear anyone.
Don't hear anyone either.
Dr. Romero, you might be on mute.
Dr. Fry, can you hear us?
Yes, thank you.
Sorry, go ahead.
Dr. Fry, go ahead. Sorry, it may have been my microphone that was not working.
Oh, thank you. I think it was. But anyway,
I was going to ask two questions. First of all, Dr. Oliver, thank you and your team for just a really great, very terrific presentation. I was going to ask the question that Ms. Foska has posed very nicely and was responded to again very nicely, and that was one that had to do with resource poor areas and race. So I'm just going to skip to my second question, which I don't expect an answer to. I asked this question at our last meeting, so I'll just pose it and you can reflect on it later. But we have spent a lot of time on this particular question of what to do about these very rare events with CVST and thrombocytopenia. And I'm just curious, again, at what point, what would trigger us revisiting this question? In other words, I know that everyone is following these cases very closely. They're being reviewed on a weekly basis and in some cases a daily basis. But at what point would we come back to this? And I'm only posing this because I'm trying to think forward about how many times we might change a recommendation. Do we revisit it when we see 2 times the amount of cases that we're seeing now? Do we revisit it when we cross the baseline number of cases? And again, I don't expect an answer. I just want to put that out there for food for thought. Thank you.
Dr. Fry, this is Dr. Oliver. I'm happy to. So I will say we plan on revisiting all of the recommendations. So everything was voted on under the EUA. So as we transition to BLA, we can reassess a larger amount of data in the setting of the pandemic then. So there is a time to kind of reassess at that point. With regards to a specific safety signal or safety issue, I would say we would do the exact same thing we did this time, which is very closely monitor. We've got the safety group meeting weekly, the work group that meets weekly. And so if there's anything that raises concern, that would go through those channels through VAST, through the work group, and come to ACIP as soon as possible. Hopefully we don't have to do this again, but I think we have shown with this really exactly how the system would work and how we would, if there's anything concerning, how we would kind of immediately bring it to ACIP.
Thank you.
This is Jose again. So let me make a comment for the general public and for anybody who's not aware of this. I'm sure that all of the members on the ACIP and the liaisons understand, but it is not uncommon for the ACIP to modify their recommendations. And that's been done before, and most salient in this is the use of the IPV, OPV recommendations that were undertaken for pediatrics, where they were completely modified over time as the risk-benefit ratio for the use of a vaccine like IPV became higher and higher. So again, this is not something that we have not done normally. So did you have any other comments?
No, thank you.
All right, very good. Dr. Talbot. Dr. Cotton?
Thanks for the excellent data and modeling, which has been really helpful to review. In addition to all the factors you considered, we should also consider the impact on immunocompromised patients. It's estimated that approximately 2 to 4% of the U.S. population is immunocompromised. And what we're seeing across numerous trials is a markedly decreased level of protection from the COVID-19 vaccines. Clinically, we're also seeing many patients infected well after 2 doses of the mRNA vaccine, many of whom have severe disease. And we are very concerned that with ongoing viral replication, the risk of producing novel variants is significant. They may be more pathogenic, they may be more transmissible, or they may even have resistance to vaccine-produced immunity, which could then have an impact on the entire community and our ability to defeat this pandemic. So in my field, there's a lot of interest in providing either booster doses or alternative vaccine administration. I've heard that this is being done in other countries such as France and Israel, or at least is being considered. Although for now in the United States, for equity, until everyone who wants a vaccine has gotten their vaccine, we've put a pause on our ability to give additional doses of vaccine for this vulnerable population. So in order to best protect this population as well as the general population, I would consider the impact of moving forward with vaccination as rapidly as possible so that we could then reach the point where we could provide additional vaccines for this vulnerable immunocompromised population. So I'd just like the committee to think about that and take that into the equation.
Thank you very much. Dr. Talbot.
Thank you. Sorry about that. I lowered my hand instead of hitting unmute. So I have a few things, and I'll try to be very direct. My major concern about talking about risk is that people are confusing the risk of this adverse event to those of oral contraceptives. And it's not the same risk because it's not necessarily the same type of clot, and it's not the same type of treatment. When women present to the emergency department with a clot, all of the protocols and all of the nation and all of the hospitals say start heparin. And that will put these patients at increased risk. And so I don't think they are the same risk. And it shouldn't be compared to OCP. So that's my one thing if you're going to advise about risk, but make sure we make it very clear that this is not a lead clot that we treat with heparin. Two, I think we don't have individual risk factors. What we have is age and sex. And so I think it's really important that when we talk about this, that we do include gender in addition to age. I don't think that we can ignore the fact that the majority of the cases have happened in women. And I also want to reiterate that women often present to emergency rooms with vague symptoms like headache or nausea and vomiting and get sent home and are not taken seriously. So I think that also needs to be remembered when we talk about the risk of this. To Dr. Cotton's comment and some comments about those that are homebound, we have seen in the recent nursing home outbreak that it is incredibly important to vaccinate those around the immunocompromised and those that are homebound. Everyone is worried if we don't have a one-dose vaccine to take into the home, we leave those patients at risk. But I do think we need to really educate the public that they are in contact with someone who is homebound or immunocompromised, that they themselves need to be vaccinated to prevent the risk of spread and an infection. And then lastly, I believe it's the Indian Health Service that has managed to keep, and I could be wrong, I would love to hear them comment. They have managed to keep all 3 vaccines at the same clinics and have done just fine. I think we're Americans. I think we have a lot of ingenuity. I think we can have multiple vaccines at one place. I think we can operationalize this. And I would hate to think that would become an insurmountable obstacle. I think it is more difficult, but I don't think it's insurmountable. And I think we can overcome this, and we can have multiple vaccines, and we can operationalize it. What I worry we can't do is educate the entire medical force and every American about the risk of this with enough detail and enough understanding so that a person can determine the risk benefit for themselves. Thank you.
Thank you. Dr. Daly.
Yeah, thanks so much. So
Dr. Daly, are you speaking?
Yep, sorry, can you hear me now?
Now we can hear you.
All right, sorry about that. So I think it is possible that we're underestimating the benefits of vaccination here, and I just want to highlight that there's at least 2 factors, and there's probably others that may lead to an underestimation. One is the spread of variants that are more easily transmissible and possibly more severe. You know, that would just only increase the likelihood of disease in the unvaccinated. And then a second is that the models that Dr. Oliver presented don't account for longer-term morbidity and mortality from COVID. There was a report out this morning, I think from the VA, that showed that those who had COVID, even who weren't hospitalized, had increased risk of mortality in the 6 months following their their COVID infection. So I think we just need to factor that in. And then I also think that if vaccine sites are able to publicize which vaccines they have available, then the public may be able to choose where to vaccinate. I realize that's a challenge because then the public's going to need to be aware of what the recommendations are, but I think that's another potential solution in contrast to having 2 vaccines on site at a given clinic. So with all those considerations, I would favor the 4th recommendation here. Thank you.
Thank you, Dr. Daly. Let me go on to Ms. McNally.
Thank you. I am in favor at this point of option 4. I agree that there are important benefits to the J&J vaccine, and I also think it's very important that people have the right to choose. What I am concerned about is that there is a very rare risk, and we would be warning for those people who may have this outcome. As I look at the EUA of Medical Products and Related Authorities guidance document dated January of 2017, I note that there is a section of this document that talks about information for recipients. And it obviously talks about the importance for people to receive a fact sheet. And I'm trying to balance that against our charter for the ACIP and our description of duties, wherein it indicates that for each vaccine, the committee advises on population groups and or circumstances in which a vaccine or related agent is recommended. And the committee also provides recommendations on the contraindications and precautions for use of the vaccine and related agents and provides information on recognized adverse events. And I guess I think that this option 4 really accomplishes that, that need for us, and it balances the role of FDA and the role of CDC. And I'm curious about my colleagues' comments on my comments. Thank you.
This is Amanda. While we're waiting for comments, and thank you, Ms. McNally, I do just want to acknowledge that people can sign up for individuals who do sign up online for vaccination. In many places, including at vaccinefinder.org, you can actually put in the vaccine that you're looking for. So we do have tools available online and can ensure that, at least on the government websites where we have those tools, that information about this risk is clearly delineated on those web pages. That does not obviously account for individuals who who do not have that technology or ability to see that, but there is an ability to identify which vaccine a person would like to choose.
Thank you. I see Dr. Long's hand is up and Dr. Sanchez's hand is also up. In an attempt to give everyone a chance to say what they need to say, I'm not ignoring you. I'll just come back to you at the end, but I want to make sure that everybody has a chance to speak here. Before we leave the voting members, are there any voting members that want to make a comment? Just say so. Okay. Let me just add my comments to what has already been said, and I won't repeat a lot of this, but I'll tell you what things that I've thought about. I mean, this is a very rare event, or very rare events, and taken in the context of all the other issues that have been discussed, other factors that are weighing in my decision are the issues of, and as Dr. Daly began to touch upon, these other risk factors that we yet have well to understand, and things such as neuropsychiatric problems associated with their infection, the long hauler syndrome. Those issues are also important, and we don't have data on that. And so limiting the vaccine to a greater population certainly would have impact on that. The issue of individuals returning for a second dose is one that's important to me. In a recent MMWR that looked at doses given between the start of the immunization drive into the middle of January, 3.4% of the population surveyed did not return during the window. I can tell you that data for my state, as of yesterday, was 11.1%, 3 times that. So clearly removing a vaccine that can be given as a single dose and is a preference among Latinos in our communities would be of detriment. And so I think that these weigh heavily on my decision. And as has been said before, I am leaning towards the number 4, the option number 4. So let me open it up now to the liaison members, and I see Dr. Dushin is top on the list.
Thank you, Dr. Romero, and to everyone who has contributed to these fantastic analyses and presentations. My question is about the model, and I think it's clear that both on the population health level and the individual health level, the benefits of this vaccine justify the risks. And this is an incredibly important vaccine that we need in our armamentarium, as Dr. Romero just described. My question is, the risk-benefit calculation is based on the current U.S. exposure risk. And over time, I believe, with these effective vaccines, the risk will recede. And I'm wondering what type of additional information we can get that will help us better understand where the disease incidence, at what level of disease, at what incidence level, this risk-benefit equation will change significantly to help guide our future use of the product. Thank you.
Rachel, do you want to comment on future use of the models?
Sure. So the models have been used in previous ACIP decision-making and have been updated as additional data become available. That's a process that we're happy to continue as we learn more about what future incidents will be. It's also why we included 2 scenarios, both that low and that moderate scenario that were included in the population level models. And so I think that we're happy to continue reassessing and providing that evidence for ACIP.
Thank you. I think that will be incredibly useful as the risk of COVID-19 recedes.
Thank you. Dr. Gluckman, please.
Thank you very much, and appreciate Dr. Oliver's thorough presentation. I would just refer folks to slide 51 of Dr. Oliver's presentation, where it puts an excellent perspective on the risk of TTS versus the potential benefits. I think that option 4, while it's important to make people aware of the risks of TTS, while that information is being provided, it is essential that people are also provided with the overwhelming benefit. For every case of TTS occurs, there are still 12 deaths prevented, 127 ICU admissions, 657 hospitalizations prevented. And then, as people have already commented on, the potential risk for long-term sequelae of COVID as we're learning more. I'm concerned that without a stronger statement from ACIP, that a decision could contribute to vaccine hesitancy, and that any contribution to vaccine hesitancy is far more likely to cause real harm to people. So while yes, information is important, we should be making a very strong and unequivocal statement about the overwhelming benefits, and recognize that there is some issues around feasibility in certain populations. Of course, when people have the opportunity to have choice, they should have choice. But I think the overwhelming risk-benefit analysis here is get the vaccine you can get.
Thank you. Dr. Shaw.
Thank you, Dr. Romero. I'd like to begin by thanking Dr. Oliver. Dr. Oliver, you gave a clinic today on the optimum way to analyze and communicate about a difficult public health policy question, and I commend you for your clarity of thought and communication. It was outstanding. I'd like to address or think about what has already been noted, which are some strands between the differences between, Dr. Oliver, your option 2 and option 4. even option 4, well, there are 2 observations I'd like to make. Both options, as your slide 71 notes, provide flexibility of choice as well as allowing receipt of this vaccine among hard-to-reach populations. I think it's important to note that believe that both options, even though it's only noted explicitly in option 2, both options would entail an official recommendation being appended to the EUA, as our colleagues from Janssen noted. And so with that, I suggest, or I posit that there's not so much of a difference between option 2 and option 4. It may come down to our understanding collectively of the language of may choose. So long as we understand that may choose is at the population level, as some commenters have noted, it may entail choosing when someone is making their appointment or at the time they decide to go get vaccines. I think at the practical level, most sites will not be able to offer 2 vaccines. In our state here in Maine, I'm not aware of a single vaccination site that simultaneously is running 2 vaccines at once. When we adjust for that understanding, I don't know that there is much of a difference between option 2 and option 4. The choice here, which I believe in retaining, comes at the time that someone decides to seek a vaccine rather than at the point of care or point of service. That's my understanding, though, Dr. Oliver, and I'd love your perspective on that.
First of all, thank you. That was very nice. I actually agree, and first of all, want to reiterate that your first point that absolutely all of the recommendations are in the context of FDA's warning statement. So we were highlighting it for that second one, that it's not necessarily going back to what we had before, it's with the setting, but yes, that any kind of future recommendation for use will be in the setting of the FDA's warning statement. And agree that in many instances it will be, up to the jurisdiction exactly how that kind of may choose another vaccine gets implemented, but very likely in many instances would be, an individual person signing up for which vaccine appointment they're going to take, which just reinforces the importance of, the public, clear communication that we've said if this is the choice ACIP will choose, that CDC will work with colleagues at disseminating.
And sorry, apologies for honing in one more time, but that option of choice already exists and is baked into option 2 as well.
Yes, correct. I think it's my interpretation from the work group's discussion, and I'm happy to, after this, kind of defer to Dr. Bell if she wants to comment again. But my interpretation was it's really option 4 is ACIP reinforcing the risk in this specific population, but that, in many situations, the benefit may outweigh that risk. Dr. Bell, do you have anything else that you would like to add for kind of work group summary?
Thanks. I guess I would say to reflect to the extent that I can reflect the work group's sense and my own opinion, I think that the issue is that we think it's important to reflect both truths, both the fact that the benefit risk is, in favor of a large benefit, but also that there is a very small risk of a serious adverse event, which people have should be aware of. So I think the idea is to reflect both truths in the language of the recommendation, to fully acknowledge both the benefit and the risk. And I think that is a little bit different than option 2, which wouldn't reflect, I don't think, in the same kind of way, the ACIP's recognition of both and the opportunity for people to make an informed choice. So that, I think, is perhaps it's a small point when you come right down to it, but I think that's essentially where it was coming from.
Thank you very much.
Dr. Bell, since you have the floor, I see that you also had your hand up to make a comment. Would you like to continue with your comment? her question.
Well, yeah, I was going to just, sort of provide my own perspective on kind of where I've come down in terms of how to approach the situation here. So I'll just make a couple of points and try not to be too verbose. I think, first, I think it's very important to recognize that this is a recommendation and a policy for now and for the current situation, which includes is not at the moment with COVID-19 at bay and things quiet and all those lovely things that we can hope for in the future. So this is a time where we need options and we need flexibility. And that's very much part of my thinking. I think the second point is that the benefits do clearly outweigh the risks from a population perspective and an individual perspective. And I think it's reassuring to me anyway that while we don't have all the information, I think we do have enough information to move forward. But at the same time, I think that there is a real risk. It's admittedly an extremely small risk and smaller than many other risks that we choose to take every day. But nonetheless, there are alternatives, at least, for us in the United States. And for that reason, that's, as I just mentioned, why I personally do prefer wording which explicitly recognizes the ACIP's recognition of both the risks and the benefits. Now, it's a little bit of a paradox that here, there's been a lot of discussion about vaccine hesitancy and what do we do about that. And my own feeling is that that's just an unknown, and we do the best we can. It's a paradox that by recognizing and detecting and acting on a very rare risk, which should in fact inspire vaccine confidence, perhaps it has the opposite effect. But in any case, I think that going forward, and this is something that a couple of people have already raised, I think that the success of this policy recommendation, whatever it is that we end up, but especially if we choose this last option, is, and also to support public confidence, it really rests on education and communication. And it's really critical that CDC, that the state and local health departments, that our partners really step up and take this task at hand and really work to make people understand what the choices are here. Thanks.
Thank you. Let me apologize, Dr. Goldman. I jumped you to go to somebody else. Please, if you have questions or comments.
Thank you. Appreciate it, sir. I'll make my comments brief so as not to reiterate everything that's already been said, but I'm a firm believer that the perfect should never be the enemy of the good. I think the risks and benefits of this vaccine clearly show that we're preventing tremendous disease and that we need to be cognizant of that fact, because to not continue the use of the vaccine would be detrimental for the overall vaccine program and preventing tremendous amounts of death, hospitalization, ICU care. But I did want to point out just a few concerns from the private practice and primary care perspective. Certainly, you would think we would be able to at least stock one vaccine, let alone two, but what is happening on the ground floor is that many of us cannot even get any vaccines to give out to any of our patients. So if we do not even have the option of a single-dose vaccine, it is going to tremendously affect the ability of vaccine distribution for many in private practice and primary care, number one. Number two, many of my patients are expressing a preference for the vaccine for a single dose because they cannot take off from work for 2 visits. They do not have the access to the care. There's certainly that health equity piece that we must be extremely aware of. So I'm greatly concerned that if we do not continue with the vaccine, it is going to dramatically reduce the ability of many patients to get vaccinated. So I also firmly believe in patient choice, and that I do think option 2 keeps it as simple as possible to make sure people can have that choice. They'll know what the FDA recommendations are and be aware of it. And my last point is that we need to be very careful in the wording that we use, because what this pandemic has shown me is how many jurisdictions will misinterpret guidelines out of convenience. And I point to the example of when we said that up to 6 weeks you can give a second dose. Many jurisdictions split the difference and just did 4 weeks across the board for both vaccines, even though that's not what the recommendation said. So I do think we have to be very careful in the words that we use and how we recommend it, as many jurisdictions might misconstrue that out of convenience as opposed to what's best for public safety and public health. Over.
Thank you for those comments. Dr. Freihofer.
Sandra Freihofer, liaison for the American Medical Association, but I'm speaking as a practicing physician. And as Dr. Goldman mentioned, I think it's very difficult for private practices to carry more than one vaccine at one time. I am very concerned about vaccinating homeless, homebound, the incarcerated, vulnerable populations, and we really do depend on this one dose and you're done vaccine with the easy storage to reach those particular populations. I'm very concerned about the variants and was impressed with the presentation by the company about coverage of this B1351 variant. I'm also concerned that a lot of these vulnerable patients and vulnerable populations would not be able to get a second dose of vaccine and might not return for that. So feel very strongly that we need to have this vaccine available. I also feel very strongly that patients should have a choice, and I was very encouraged by Dr. Cohn's statement that there is a way on the vaccine, looking for a vaccine that you can determine a place where you can get the particular vaccine that you want, because some people really care about which vaccine they're getting. Some people may not be as interested. But I was wondering about, and I liked what Dr. Bell said about the two truths, wanting to, and the reasoning behind her choice of option 4. But I share Dr. Shaw and Dr. Goldman's concern that it's just a little complicated and the wording makes it sound a little shady and might be discouraging, and I worry about increasing vaccine hesitancy. And I asked Dr. Cohn, if you had the reaffirmed recommendations for all sex, age, and sex, and if you had that FDA warning in the EUA, how would that change the outreach of the CDC to make sure that practitioners, that clinicians, that the public understood the risk and the side effects? Because it's just a much simpler way of expressing the same thing. And I didn't see really much difference when I thought about them.
Dr. Freihofer, CDC would develop, if for either options 2 or 4, CDC would develop the same tools and communication and education products to help women and providers understand the benefits as has been recommended, as well as the risks.
Thank you.
Thank you.
And thank all of you for a great presentation today. It's just, I'm so proud of the hard work and all the detail and research that's gone into making sure that our vaccine safety surveillance system is looking after the American public.
Thank you. Dr. Zahn.
Thank you, Dr. Romero. And I'd like to echo others' comments and appreciation of all of the outstanding presentation and conversation today. And Dr. Freihofer actually touched on it, but I guess I was following up on Dr. Shaw's comments about the difference between option 2 versus option 4. In public health, we're anticipating that we're all anticipating over time the mass vaccination events and the pods we set up where hundreds of people show up, those are going to go away. And at that point, for public health, from our side, we, with a lot of community provider assistance, really anticipate reaching out to communities, reaching to them where they are. Obviously, the homeless population is an obvious group that will need this outreach. But when we're reaching out, option 2 versus option 4 would be really quite different. If there are potential groups who would not be indicated or would not want to have the vaccine, then you potentially have to bring 2 along. That would be logistically difficult. And I think more than that, a lot of outreach partners that we're relying on that have done such great work have been first responders, volunteer nurses, People who do a great job vaccinating, but I'm not sure are going to be tremendously comfortable talking through the risks and potential pluses and minuses for women about the issue when they may be the first person to talk to them or the first medical provider to talk to them to broach the issue that they need to be vaccinated. So certainly understand the issue. This is not simple, but if you add the wrinkle of women under 50 be aware, and they may choose, then the logistics of those outreach. Can we do it? Yeah, absolutely. Does it become more complicated? Yeah, it definitely does. So.
Thank you. I see Dr. Lee has her hand up, and I don't believe she said anything in this session yet. So, Dr. Lee.
Thank you so much. I'm going to start with a slightly long preamble, but I promise I'm going to get to my point, which is, number one, clearly at a population level, the benefit-risk balance favors vaccine use. Even in the last 11-day pause, what's striking to me is that there have been deaths and ICU admissions that would have been potentially preventable had we not done the pause. At the same time, though, I recognize that the last 11 days, to me, have been reassuring in that we haven't identified hundreds more cases despite enhanced awareness and case finding across the U.S., which was actually my worry last week. We have a lot better information about the risk estimates by age and sex, and I think that can inform our decision-making today. So just in sum, last week I would have made a different decision than what I would advocate for this week. From an access and acceptability standpoint, I just want to state that, ACIP's role, I think, in this instance should be to ensure access to vaccine. Individuals should absolutely be well informed, as they always should be, and they should have the ability to make an informed choice and encouraged to talk with their healthcare providers if they wish. But that acceptability is not just a J&J issue, it is a much broader issue. So I think that we cannot do enough to support and ensure that we are always educating patients and the public, and we need it on all fronts. So I don't actually think this it is a very specific issue, but I feel like it is a much broader issue that we need to address. On the implementation front, I just want to state that, we are going to have these issues all throughout. Right now, we have one vaccine available only for 16 to 17-year-olds. So in order to be able to ensure access, we are functionally carrying multiple products, maybe not on the same day, but many sites are doing this in order to ensure access to 16 and 17-year-olds because we only have one product available currently in the U.S. That goes down to that age group. And we do have vulnerable children in that age group. Finally, from a communication standpoint, I want to emphasize the point that was made earlier that this is not just risk communication, it is benefit risk communication. And that similar to what we've done for anaphylaxis and making sure that people have the tools and the communication and the sort of screening available at vaccination sites, I kind of think that same approach needs to happen here, and would also just make sure that everything that we're doing incorporates health literacy principles and multiple languages to ensure that we are truly informing everyone who may wish to have the vaccine. But I'm going to actually now take a different stance, and I did not at all in the beginning of today think I was going to take this stance. But from a process perspective, I actually think we need to think really carefully about the implications of what the recommendations will be. So for me, I'll just state at the outset that number 1 and number 3 are off my list. I've been thinking carefully and listening to the arguments for 2 and 4 here up on the slide, and having spent a lot of time with that evidence to recommendations framework, I do not think that 4th recommendation falls within what we would normally consider as a recommendation per our usual framework. It really should fall under 2, reaffirm recommendations for all age and sex, with the same comment about the FDA warning and all of the information that I think is in 4 absolutely needs to happen. I was feeling at the beginning of today that I needed all that additional information in 4 to feel comfortable with any recommendation as a way to state sort of our concern about the benefit-risk balance. But I have to say that I think it's going to be actually extremely confusing because every recommendation we make is a benefit-risk balance. And we cannot parse out every single benefit and risk in every single recommendation for every single vaccine, COVID or non-COVID. So I actually think we need to, from a procedural standpoint, go back to number 2 and make sure that all of the discussion points that were brought up for number 4 are incorporated in the way we implement, but that the recommendation should be consistent with the evidence to recommendations framework. And I would love to hear comments about that.
Thank you very much. So to everybody who still has their hand up, and to those that want to make a comment, we will try to get to everyone. I want to remind you that we're scheduled to end at 5 p.m. Eastern time. We could go over, but really want to stay to that window. So we'll go on to the comments. Let me see here. Dr. Grog.
Yes, I'll keep it simple, representing the American Osteopathic Association, but I have an immune deficiency and did not respond to the vaccine. I think that we have caused a lot of vaccine hesitancy in our pause for the Johnson & Johnson, and as others have stated, it's been some deaths in ICU and hospitalizations and long-term sequelae and things that have probably happened over the last week. I would like to see, I'm confused whether it should be 2 or 4, but I'd like to see everybody vote forward on the J&J vaccine and heavily inform our healthcare providers about insulin and other things, and let's get this done. Thank you.
Thank you. Dr. Hayes.
Carol Hayes with the American College of Nurse Midwives. I've been working in a mass vaccination clinic, and I want to reassure you that we have carried all 3 vaccines, and we have little tents set up, and where our little cubicles are numbered, and somebody in the back room draws up all the vaccines, and I just sit there and jab the arms. And it really is possible to stock 3 vaccines and to organize at 3 vaccines and to have people choose when they walk in the door which one they want. And I also want to just thank everyone who's presented all the data today. Everyone did such a fabulous job. And I also want to reiterate what Dr. Lee just said, that I feel as though at the beginning I was going to be a number 4 girl, and now I'm a number 2 girl. So thank you.
This is Amanda. I'd like to I'm going to ask Dr. Oliver to put some additional language, listening to this discussion about 2 and 4 for people to consider. We have a lot of people whose hands are raised, so this will just give you one more thing to comment on, as Dr. Romero said very briefly. But I'm going to hand it over to Dr. Oliver. Are you ready? I don't think, yeah, just say it and then, yeah.
So we'll work on getting these to where we can project them in just a second. But the language that was voted on previously says the Janssen, which would be option 2, the Janssen COVID-19 vaccine is recommended for persons 18 years of age and older in the U.S. population under the FDA's emergency use authorization.
And then the proposal would be for that, with that language in the recommendation, we would then describe the risks and benefits and in the clinical considerations section, but have the overall recommendation point to the EUA only. So it's essentially 2 with the additional language pointing to the FDA emergency use authorization.
Yeah, go ahead.
Yeah, and Sarah's going to repeat the current language for number 4.
So for an option number 4, the vote would say the Janssen COVID-19 vaccine is recommended for persons 18 years of age and older in the U.S. population under the FDA's emergency use authorization. Women aged less than 50 years should be aware of the increased risk of thrombosis with thrombocytopenia syndrome and may choose another COVID-19 vaccine authorized for use in the United States.
And I think since we haven't heard anybody supporting 1 or 3, we're going to send these 2 language options in a slide, and we're going to project those up for people to review.
So we're going to take some comments while they finish the slides. Dr. Eckert, please. Dr. Eckert? We'll come back. Dr. Alt?
I don't see a huge difference between 2 and 4, if depending on the way the warning statement is in the EUA. It looks like it would have to duplicate a lot of the information we already have in 4. Does the language for the warning statement exist somewhere? Is there some way to look at it, or does it need to be developed still?
This is Dr. Oliver. It was presented at the very end of the Janssen slide. When we get through our slide stuff, we can we can show that again.
Thank you.
This is Dr. Eckert. I'm sorry that there was a leaf blower right outside my door right as you called on me, and I thought, oh my goodness, you will only hear the leaf blower. So
Please go ahead.
But I just want to say, as an individual here, not speaking on behalf of ACOG, this is my individual thoughts, but my concern is that number 4 does potentially enhance obstacles to vaccination. And we have definitely seen that individuals who are pregnant have met frequently with obstacles. And I question whether, since the CDC will and has been doing a great job with information and information sharing, if doing that for number 2 and number 4, like Dr. Alt just mentioned, if they're really all that different. And so I personally would advocate for having the fewest obstacles as possible in the way of vaccination and going with number 2. Thank you.
Thank you. Dr. Weiser.
Hi. I just want to comment on the fact that Indian Health Service arguably serves some of the most remote and hard to reach populations in the country. And in our jurisdiction, we've been receiving all 3 vaccines and providing all 3 vaccines. And some of our sites are actually handling all 3 vaccines. The Pfizer just for the 16 and 17-year-olds, the Moderna for the majority of the adult population, and then the Janssen vaccine, which some people were waiting to receive and not choosing to get the other one. So as part of a health system, I think we have proven that we can handle all 3 vaccines. Typically, when we have a large vaccination campaign or site, we'll give mainly 1 vaccine on that day if we're doing a drive-up or a large-scale vaccination event. But we have the ability to keep the vaccine straight and make sure that people get the vaccine that they've asked for. Thank you.
Thank you. Ms. McNally?
Thank you. I just want to confirm that the FDA agreed warning and precaution regarding thrombosis with thrombocytopenia that the Johnson & Johnson presentation showed is what would appear in the EUA fact sheet for recipients.
Would somebody from the company please respond on that question?
Yes, this is Dr. Waldstreicher. That was the fact sheet for healthcare professionals, and the one for recipients is different, but it's very clear, and it gives information for recipients about the signs and symptoms and to present if they have any of the following of those signs and symptoms, as well as describing the overall event as well.
I find this to be very relevant to the discussion, and wondering if it would be possible to see some of this proposed language. And I mention it because of the fact that informed consent is not required for a product authorized under EUA.
Excuse me, this is Dr. Bell. Would it be possible for FDA to make a comment about what is going to be in the fact sheet?
Yeah, hi, this is Doran Fink. I think it would be best if Janssen has the ability to share the language for the recipient fact sheet for them to do that.
Dr. Marie, you have that in front of you. Would you be able to do that?
We can also continue on with comments and give you guys a few minutes. If you'd like to send something, we can project it.
Yeah, I think that would be good, so we can move through these.
Thank you.
Let me go back to the top of the list and work my way down again. Dr. Long, please.
I agree with so many things that people have said, especially Dr. Bell and Dr. Lee. I think we shouldn't talk about this as very rare or, you can't, I take it all out of all medical writing. Rare is rare and it's difficult to quantify. If we're talking about the age group of women that we're talking about, it's 1 in 140,000. Now, some people would say that was rare and some people would say, I would never say it was very rare. The other thing is that people perceive and want to take risks at different levels. So I think we want to be sure that we do allow choice. The next comment is because it's difficult to do doesn't mean that we can abrogate our responsibility to protect the public as well as individuals. I think if we had 3 vaccines, they were all single dose, they had the same efficacy, we would be preferring another vaccine, and they were all available. We would be preferring 2 other vaccines in this age group. I certainly would be preferring that. And I don't say that we should say that specifically now. I think we want to reaffirm that the vaccines themselves, the benefits outweigh the risks. Or the next thing is that you ought to receive any vaccine you can get as soon as you can get it. And then we have to have a different way to say number 4, because number 4 to me kind of comes out as a petulant young woman rather than we're experts. We recognize this risk. This risk may be more important to some people, or maybe a single injection is more important to that young lady who's choosing between vaccines. But to say may choose, I don't think it's saying what we recommend. And I'm not for just leaving the FDA warning, because the FDA does the licensure and the legality part of it. We're supposed to nuance that by all of our deliberations to protect the population as well as to protect the individual. And I think that the benefit outweighing the risks in that younger age population of women, I don't think that I buy that, that they won't otherwise be vaccinated. So I think we have to do justice to both truths. And so I think the way you're using the words now are good. I just think we need to have something better than may choose.
Thank you. Dr. Sanchez.
No, thank you. I agree with Dr. Long. I actually had a question and then a comment. When you look at the list of women, and remember one was a man who had the thromboses. One of them was 59, and I know that the risk benefits and the risks were all greater than 50, but any consideration of age 60?
This is Sarah. I think the work group, we tried to look at what was known with the current age distribution. We had to make some assumptions when we started doing this work a couple of days ago. And so the work was done with the 50-year-old cutoff. I think we did look at information. So the work group saw more slides and more information than what was presented publicly today. There was some information presented for 65 and older, so not 60, but 65. Ultimately, the issue is the population 65 and older that's remaining to be vaccinated is very small, or is much smaller than the 50 to 64-year-old population. And so some of that definitely impacts the modeling results when it comes to that. But we did not specifically look at a risk-benefit analysis with the line drawn at age 60 years.
Because when we, the data as of April 14th, among the cases, the range was 18 to 59. And I know that it's, it's just some cases. But, because, I fully understand the benefits of immunization. We cannot argue with that. But I also understand the risk, and the risk is not it's not an insignificant risk. It makes me nervous. And I think that that at least with this vaccine, yes, there's only one male during the trial that had it, so it doesn't seem to be zero in men, although, yes, quite rare, or quite infrequent. And I just, I think that the choice, as one of them, as the number 4 is worded, it sounds almost like the choice should only be given to women and not to everyone. And so I'm a little bit hesitant about that one as well. But I'm not sure if 50 is the age cutoff that I would use.
Thank you. Dr. Paling. Dr. Paling? We'll come back to Dr. Oh,
thank you.
Go ahead.
I'm here. Thank you. Ms. McNally shared her thought process of how she was thinking about this and put it into context and asked for a response. I wanted to respond and affirm that I am thinking along the same lines and appreciate her adding clarity to this thought process. I also wanted to say thank you for getting the exact wording for the public, because that is a very important part of the consideration for me. Thank you.
Thank you. Ms. Hayes.
I apologize. I forgot to take my hand down.
Thank you. Dr. Holly? Dr. Howell?
Oh, hi. Thanks. Molly Howell, representing the Association of Immunization Managers. I just wanted to say that yesterday we surveyed our members, who are the 64 awardees implementing the COVID vaccination programs in the United States, and 81% were supportive of, I would say, either option 2 or option 4. So I just wanted to share with that, and that we will work with either. Thank you.
Thank you very much. Dr. Stinchfield.
Yes, thank you, Patsy Stinchfield, representing NAPNAP. Personal thoughts here from the state of Minnesota that is seeing all the variants and increasing numbers, and as we speak are accepting young adults into our children's hospitals because of very high census across the state with the disease. So what makes me nervous is the climbing incidence of COVID. I have an urgency to move on this, what our former option 2, which I think we'll now call the top box. I would support that. And I think because it's most reflective of the data that we've seen today and the conversation, I think it is will have the least unintended consequences for those who are reading it. And I do think in a hospital that's given 20,000 doses so far, 2 products, it is very doable well to have two different products. For example, our Johnson & Johnson, we've been very happy to give to 18 and over young autistic males who are combative. They're under a sedated procedure, and it's worked out really, really well. An already planned procedure. We co-administer that vaccine with very happy families at the end. I think, we can do these on different days, different sites, different locations. So that implementation, I don't have any concerns about that. I do think that in terms of vaccine hesitancy, yes, I think this pause has made people nervous, but I think in the end, time will tell. But because of the excellent work, and Dr. Oliver, what a masterclass you gave us today, this will help increase the confidence in the United States and across the world. And I'll just end by saying that I do have a considerable concern about our global problems right now, looking at India and Brazil, and the urgency, again, not only in the U.S., but across the world, to use all the tools that we have to get the pandemic behind us. So I feel very comfortable in that top phraseology with the details in the EUA. Thanks.
Thank you. Dr. Whitley Williams.
Yes, thank you. Pat Whitley Williams, representing the National Medical Association. And I also thank you, Dr. Oliver, and your group for just outstanding presentations. Our preliminary considerations would be supportive of reaffirming the recommendation for use of this vaccine. It's well recognized that persons in the Black and Brown community have been disproportionately affected by this pandemic. So the benefit of a vaccine that is effective far outweighs the risk, and especially in hard-to-reach vulnerable populations. And I also concur with what Patsy Stinchfield just said in terms of the global impact. But also would encourage, and I know this is work that's ongoing, to really try and get to the bottom of this underlying cause of the TTS, and particularly as it relates to age group, gender, and race ethnicity. Thank you.
Thank you. Dr. Arthur.
Thank you. Phyllis Arthur on behalf of BIO. I actually just wanted to suggest that whatever the decision the ACIP voting members make, this is an excellent opportunity to recommit to getting all the vaccinees to use BSafe. We think this is an extremely important tool for helping people pay attention to their side effects, report them, and gather the really important data on vaccine safety that's so important to our overall efforts. We've had many reports of people not being told about BSafe when they get vaccinated, although other places are doing a great job. And this is an opportunity, given any need to change the daily information people get in the text, to actually encourage more and more citizens to enroll and fill it out. So just wanted to put a word in for, as we look at BSafe, having another wave of encouraging people to use it.
Thank you. Dr. Bernstein, I'm going to skip you because you've already spoken. I don't think we've heard from Dr. Chen. Dr. Chen, and then I'll come back to Dr. Bernstein.
Yes, thank you. And so I've been really enjoying the discussion, and I think I agree with what Dr. Shah initially kind of articulated, which is that in essence, box top and box bottom are not significantly different from each other, as long as we understand that in the implementation that we would not, have a formal, complicated consent process for the be aware or making aware statement, and then for the ability to choose, that we would not make it a requirement for the local point of immunization clinic to be required to store 2 or more vaccines. So I think that as long as I understood that correctly, there really isn't that large distinction between the top and the bottom box. And so I do wonder if that bottom box, having not much of a real distinction, has more of a risk for misinterpretation from local kind of smaller implementation teams and programs. I think we heard from other large programs that they have no problem with juggling, 2 or 3 vaccines simultaneously on any single day. But I do wonder about these smaller outfits. Again, in my state of Maryland, we have, individual medical practitioners and pharmacists, dentists, and veterinarians that are starting to be trained to be able to implement these vaccines. And again, I can't imagine that they will be able to juggle multiple products simultaneously. So I think that, again, as long as we saw that enhanced labeling information, and we have an aggressive campaign for educating the public and the medical community, I think in essence those boxes are not really different from each other. And to make it simpler, that top box is really what I favor. So I'll stop there.
Thank you, Dr. Lett.
Yes, hi, this is Susan Lett from CSTE. We're wondering if you could tell us a little more about implementation, will happen, because this is we've never sort of had this pause before, and sort of sequentially, can you describe it?
Dr. Lett, you're asking us to describe the pause? Oh, the implementation of if the pause is lifted?
Yes, there have been some questions amongst our members and other members that we've been talking with about how exactly things would get re-implemented as soon as we have to wait for certain language to be available, for materials to be available, I don't know about screening forms and standing orders. We're just trying to understand how that would happen.
Dr. Lett, this is Amanda. So if there is a decision in the next half hour, hint, hint, sorry, then we are awaiting and anticipate that the FDA will be putting out newly approved EUA language, which we've pulled up for you to see now, the language which we can have the company take us through that's in the patient FAQ sheet. And at that time, if the CDC director approves the decision made by CDC, we would publish that on the ACIP website and through other information sharing, and people would be allowed to presume use of doses that they have available potentially already. That being said, immediately over the next 72 to 96 hours, so Monday, there's going to be another COCA call that describes all of this information. There will be an MMWR that shares all of this information. And additionally, we will be taking the input from today along with this language that FDA will publish, and both FDA and CDC will have communication materials and education materials, including infographics, patient fact sheets that are smaller and focused just on this issue, and those will be available early next week.
Do you want to walk through this, and then I'll take the final questions that are here. So go ahead.
Yeah,
Go ahead, J&J.
This is Dr. Oliver. Yeah. We just got the recipient fact sheet. So if somebody from J&J wants to walk us through that.
Yeah, certainly. This is Dr. Waldstreicher. So this is the recipient fact sheet, and this is the section that's on the risks of the Janssen COVID vaccine. Prior to this, it first describes the side effects reported, like injection site reactions and general side effects. Then it talks about anaphylaxis and gives the symptoms of those. And then you're seeing the section there highlighted in yellow, where it talks about this new thrombosis with thrombocytopenia, but in language, appropriate language for this fact sheet. So it talks about blood clots involving vessels in the brain, et cetera, goes through those. It talks about it's occurred in some people who have received the vaccine, and in people who have developed these clots. I'm not going to read the whole thing word for word, but it talks about the fact that they develop approximately 1 to 2 weeks following vaccination, and that most people who develop these blood clots and low level of platelets were females ages 18 through 49 years. The chances of having this occur is remote, and you should seek medical attention right away. if you have any of the following symptoms, and then it gives the list of symptoms. This is consistent with professional society guidance as well. And that's the end of that section.
Thank you.
Dr. Romero, may I ask a follow-up question?
Go ahead, please.
Does this fact sheet indicate that there is an alternative, that being the mRNA vaccines?
What the fact sheet shows, and it's actually later on down there, it says other vaccines to prevent COVID-19 may be available under emergency use authorization. It's probably there if you scroll down. Under Janssen, the question is, are other choices available for preventing COVID-19 vaccine? It's up a little bit, I'm sorry. Are other choices? Oh. There it is, okay. Are other choices available for preventing COVID-19 besides Janssen COVID vaccine? Currently, there is no FDA-approved alternative vaccine available for prevention of COVID-19. Other vaccines to prevent COVID-19 may be available under emergency use authorization.
And this is Amanda. I'll just add that in the CDC educational materials, we will describe all options available, and we're able to speak about all of the products as opposed to the EUAs, which have to focus on the individual product that it's authorized for.
Thank you. The information is very helpful.
Dr. Romero, this is Dr. Bell. Can I just ask one follow-up question before we head on to the other people with questions?
Of course, Dr. Bell.
Thank you. So this EUA fact sheet is given to patients when they're getting ready to have a shot. Is that correct? In other words, they're in the clinic, they're about to get a shot, and it's the Janssen vaccine, and so they get this piece of paper. Is that how these EUA fact sheets are used?
Does someone from the company or FDA want to respond to that?
Yes, generally, this is how the fact sheet is used. That we also post it on the website, and I believe that it's posted on other websites as well, such as that would accompany the vaccine.
I will tell you in practice that there are that sometimes you receive this information through your appointment. You receive an email with the PDFs of this information as well, but it should be given at some point between when you make an appointment and you get vaccinated.
Thanks.
Sorry, I lost my list of speakers here. Let me go back to Dr. Bernstein, then Dr. Paline, and then I'm going to close it unless somebody has some very pressing comments. Dr. Bernstein.
Thanks, Dr. Romero. It's incredibly impressive to me how well our nation's vaccine safety surveillance systems work. The Vaccine Adverse Events Reporting System, VAERS, which is not designed to assess causality, identified a safety signal of real concern, prompting further investigation. As Drs. Oliver, Bell, and the COVID-19 work group have presented, the ACIP closely examines disease epidemiology, vaccine supply, implementation concerns, public values and acceptance, and equity. Its three overarching pillars of science, implementation, and equity are important. After our emergency meeting last week, I felt implementation and equity favored reinstituting the J&J vaccine, but the science was much less clear to me. The brief pause, measured in days, appropriately continued to allow additional data to be collected and analyzed. The ACIP process works in a timely and transparent way, which should improve the public's understanding of this safety signal. Today's presentations and discussions have convinced me that lifting the pause on J&J's vaccine is in the best public health interest of the U.S. population. Any vaccine may have side effects, and in this case, the risk of blood clots and low platelets, even resulting in fatality in some, has now been extensively studied and will continue to be monitored. Plus, patient education and provider recognition are expected to help improve outcomes. Important information also will now be conveyed in the FDA's EUA fact sheet, the CDC's materials, and through local public health officials. The 3 COVID-19 vaccines currently authorized in the U.S. are amazingly safe and effective. In my mind, their benefits far outweigh identified or perceived risks. We need as many people to be vaccinated as possible in order to reach community immunity and put the pandemic behind us. The J&J vaccine will help us do just that. So I prefer the option 2 or the top box, and I think that we can include nuanced language in our clinical considerations that often accompany these recommendations. Thank you.
Dr. Paling.
Can we pull up the FDA EUA sheet again, the bolded part? I wanted to look at that. And in particular, my question on that for the component to the, I was looking at the one for patients in the public. And in that yellow component, my question, I just wanted to highlight that headaches are the most common early symptom of these thrombosis and thrombocytopenia. And so I am concerned that that is buried. I think it was the 5th one down. And I hope on the CDC website we could elevate that location. And I'm happy, I would appreciate input from the FDA and the thought process of ordering. Thank you.
Go ahead.
This is Amanda. We will take that into account as we're developing our CDC materials. That's a great point. I also want to comment on something that Dr. Talbot said much earlier regarding emergency room and understanding the risk of clots, especially in this age group of women. And one of the key things that we have been communicating and we will continue to communicate in our provider education is that one of the first things that a provider should do is check a platelet count, and that should inform treatment and management. And so we are very grateful to our colleagues at ASH and other colleagues who have supported information around the clinical management to ensure that patients are treated, that this is recognized and treated appropriately early.
So before we move on to a motion, I think we're ready to discuss this. I just want to say the following. While I can vote for number 2, I think we've already begun to see in the discussion that there is a bit of selective interpretation of that recommendation. Women are not going to be offered at that site across the country an option to a second vaccine. And that's going to place certain women at a disadvantage that may decide that the vaccine being offered at that site is not the one for them. And they're going to have to shop for a vaccine. So that is an issue. I just want to point that out. But as I said, I think I can live with number 1 because it does reinstate the Janssen vaccine, which I think is essential to our vaccine efforts. So I think at this point we'll end discussion and see if anybody wishes to make a motion. Voting members, sorry. Dr. Lee.
So I move that we recommend the Janssen COVID-19 vaccine is recommended for persons 18 years of age and older in the U.S. population under the FDA's emergency use authorization period.
The top box to clarify, Dr. Lee.
The top box, yes. Thank you.
Thank you.
May I have a second, please? Dr. Alt?
This is Kevin Alt. I'll second it.
Thank you very much. Is there any further discussion from the voting members only? Let me see here. Dr. Sanchez?
Is it possible to vote between the two?
Say that again. Say that again.
Is it possible to vote one versus the other?
Dr. Sanchez, so for procedurally, the motion that has been made now needs to be voted on, yes or no in and of itself. If this motion passes, this motion carries. If this motion does not pass, we can put another motion on the table.
Is that clear, Dr. Sanchez? We have to vote on this motion at this time.
Okay.
Thank you. Dr. Long.
I will not comment further on this motion.
Thank you. Dr. Bernstein. Dr. Bernstein, your hand is up.
I meant to take it down.
Thank you. So I'm going to ask that all the voting members please turn their videos on and get ready to offer a vote. All right.
So I just want to remind our members, we are still not doing really electronic voting, but as you go through your votes, please let everyone know again if you have any conflicts of interest. And I believe there are a couple of members who will be abstaining from the vote due to being involved in clinical trials with COVID vaccines. So please indicate that. Additionally, at the end, after the vote, we will have an opportunity for those of you who would like to share and explain your yes or no vote. So we will have the opportunity to do that after we go through the members. So Dr. Romero?
Thank you. So the motion to be voted upon is the Janssen COVID-19 vaccine is recommended for persons 18 years of age and older in the U.S. population under the FDA's emergency use authorization. So I'll begin. Dr. Alt.
Alt, yes, no conflicts.
Dr. Bata. Oh, sorry, Ms. Bata.
Bata, yes, no conflict.
Dr. Bell.
Excuse me, yes, no conflict.
Dr. Bernstein.
Frank Bernstein, yes, no conflicts.
Dr. Chen.
Chen, yes, no conflicts.
Dr. Daly.
Daly, yes, no conflicts.
Dr. Fry.
Fry, abstain due to conflicts with clinical trials.
Thank you. Dr. Cotton.
Cotton, yes, no conflict.
Dr. Lee.
Lee, yes, no conflicts.
Dr. Long.
No, no conflict.
Ms. McNally.
No, no conflict.
Dr. Paling.
Paling, yes, no conflict.
Dr. Sanchez.
Sanchez, no, no conflict.
Dr. Talbot.
Talbot, no, and no conflict.
Romero, yes, no conflicts. Thank you, Dr. Cohn. So the vote is 10 in favor, 4 opposed, and 1 abstention. The motion carries. The Janssen COVID vaccine is recommended for persons 18 years of age and older in the U.S. population under the FDA's emergency use authorization. Thank you all very much. So are there any of the voting members that wish to offer comment at this time?
Please raise your hand.
Yeah. Is Dr. Bell, Dr. Bell? You can stop sharing now if you wish.
Thank you. I wanted to reiterate a theme, which is the importance of CDC and the state and local health departments and the partner organizations providing clear communication and education on this topic. I voted yes. I can live with this recommendation. But I think that under an emergency use authorization, where there is no signed informed consent, that it could be that ACIP recommendations might need to reflect some more nuanced concerns than under the usual procedure. Nonetheless, as I say, I voted yes, but I am concerned that that the consumers and women in this age group in particular will not be adequately informed just by the FDA EUA fact sheets. And so we really are depending on the public health agencies and the partner organizations to make sure that people actually are informed and are empowered and that they actually get a balanced perspective.
Thank you, Dr. Bell. Dr. Alt.
I was going to agree with Dr. Bell. I was thinking the very same thing.
Thank you. Dr. Sanchez.
I agree with Dr. Bell, and actually that is precisely the reason that I voted no. I have no problem with the continued availability of this vaccine. I think if someone makes an informed consent after having and knowing the risks involved, I'm fine, let them get it. But I think that we just making a blanket recommendation, knowing the risks that seem to have biologic plausibility and severity, I think we have to have a stronger language and make sure that people are informed appropriately.
Thank you, Dr. Palin.
Thank you. The detection of thrombosis and thrombocytopenia after 6 cases and over 7 million doses administered clearly demonstrates these safety systems are both sensitive and effective. The COVID-19 pandemic continues to cause significant morbidity and mortality. Not only does COVID-19 disease cause many complications, including but not limited to increased risk of serious clot, multi-inflammatory syndrome, post-infection neuropsychological syndromes, and long hauler syndrome. This pause has been effective in increasing awareness of the rare and serious events of thrombosis and thrombocytopenia, highlighting the importance of timely diagnosis, evaluation, and treatment, and gathering data needed for a risk-benefit analysis. At the population individual level, the benefits of reaffirming the Janssen vaccine is high and outweigh the risk. For all these reasons, I believe it is in the best interest of the public to reaffirm recommendation for this vaccine with the warning added to the FDA EUA. Thank you.
Thank you, Dr. Long.
Yes, I did not object to the recommendation. I objected to the absence of any kind of guidance from us. This is an age group that is most at risk, that is getting this vaccine predominantly to save other people's lives and morbidity, not their own. And I think we have a responsibility to be certain that they know this. And if they choose to be vaccinated with this anyhow, we want to respect that choice. But I'm very sorry that we haven't chosen to put up front the knowledge that we have that this is unique, it's clustered, it's almost certainly related to the vaccine, and there are options.
Thank you, Dr. Long. Dr. Bernstein.
I was wondering if you could clarify when language within the clinical considerations, I think some of the concerns that I'm hearing, I would assume would be written into the clinical considerations that are updated the way they were when we were talking about anaphylaxis initially with the Pfizer vaccine, and there were clear considerations that were readily available to the public, published on the CDC website and in MMWR, and I would assume that kind of content would be and language would appear in the clinical consideration.
Dr. Cohn.
Thanks, Dr. Bernstein. So this, all of this language around the risks and benefits and the decision to get vaccinated that were presented in the course of the discussion today, there's language that will still be included in the MMWR. And additionally, the clinical considerations will be updated. We anticipate that if the director agrees to reaffirm this recommendation, that all of this will be published during the day on Tuesday.
Dr. Lee.
Thank you.
Thank you. I want to emphasize that I actually agree with all of my colleagues who voted no, that I absolutely think this is a serious adverse event. We need to continue to ensure that awareness is raised. I am perhaps a procedural nerd in that I think that the only choices that we really had were to recommend or to not recommend or to do shared clinical decision making. And I think that the emphasis and the vote itself reflects the challenge that is before us today, or that was before us today, that we need to be able to reflect the importance of this particular adverse event and elevate it. But I also think that we have to come out with a clear recommendation. I think that the recommendation number 4 would have been confusing, and it's actually inconsistent with all of the other prior ACIP recommendations we've given. So I think that is why I had chosen the top box instead of the bottom box. Thank you.
Thank you. So I will exercise Chair's prerogative and have the last word. As I stated before, I was able to vote for number 1. Excuse me. I think that it states clearly that Janssen vaccine can be reinstituted and should be reinstituted. I acknowledge, as does everyone else, that these events are rare, but they are serious. At the same time, it places upon us, those that will make this vaccine available and will be distributing it, to make an informed public about the risks of the vaccine. This is not something that we will be able to simply say we're following the ACIP recommendation. It is our responsibility as clinicians to make sure that women understand this risk, and when possible, that they have an alternative at the same site that you're administering the vaccine. So I will end there. Let me again, I think the Oscar for best presentation goes to Dr. Oliver. There's applause in the room here if you can't hear it. Thank you all for your time. Thank you all, that is the voting members, the liaisons, the ex officios. I don't think I'm exaggerating when I'm saying that I think there's no group that has put in as much time as you have. I thank everybody else in the room. Oh, I'm sorry. Forgive me, Dr. Wharton. You're behind me. I didn't see you.
I just wanted to add my thanks to those of Dr. Romero. I know this has been difficult.
We can't quite hear her.
I know this has been difficult and it has taken a lot of time, and we greatly appreciate your thoughtful deliberation.
Is that clear enough for you? Can you all hear that? Why don't you come down here, Dr. Wharton? I think this is important that they hear this.
I just wanted to say thank you to all of you for the time you've put into this, thoughtful consideration, the deliberative process you've engaged in, and your coming to a conclusion on this important matter for us. We really appreciate your support and help, and thank you for everything you've done to support the COVID vaccine program.
Thank you. And again, thank you, Jessica, Dr. Cohn, Stephanie, for everything you're doing. And I'll turn it back over to Dr. Cohn.
Before we close the meeting out, I think just to reiterate, Dr. Wharton, there have been probably hundreds of people working in our immunization safety office and at FDA collaboratively on all of this work over the past 2 weeks. So thank you. I just want to remind everybody that we do have an ACIP meeting scheduled for May 5th to discuss dengue and rabies vaccines. Dr. Oliver is off the hook for that one. But we do anticipate if there is a future authorization by FDA, we do anticipate having an additional ACIP emergency meeting to review any additional authorizations for additional age groups for vaccines in the near future. So stay on the lookout for updates. Thanks.
So with that, then, I will knuckle us out of the meeting. Meeting adjourned.